Morning, everyone, especially those that are living proof that you can start speedrunning Rakia at 1 a.m. and then still be in the front row attending the conference this morning. Today's first speech is, you just mute the computer. All right, today's first speech comes from the NCC HPC Montenegro. It is a bridge between scientists and researchers and largest supercomputing, high-performance computing resources in Europe. It is a joint project between University of Donja Gorica and University of Montenegro, and they are not only providing supercomputing resources to Montenegrin researchers, but also providing help with accessing this resource and also education on how to utilize it in the best possible way. It is my honor to announce Tomo Popovic from EuroCC.
Thank you, Milutin. Thank you, everyone. I first want to welcome you to Montenegro. It is always an honor to host an event such as this one. It is not very often that we have over 300 people from all over the Europe or all over the world to visit Montenegro, especially Cetinje, which I personally believe you enjoy. It's a beautiful little town with a lot of tradition and history. I want to thank Eastern European Machine Learning Association, Petar, his colleagues. I want to thank Montenegrin AI Association for co-organizing this event and probably being persuasive to the group to organize it here in Montenegro. And I want to thank you for the opportunity to partner with you guys and provide as little or as much support that you asked from us to support. I'm wearing a couple of hats here, but the main hat here is HPC National Competence Center of Montenegro, and also I'm coming from University of Donja Gorica, and here we also have representatives from University of Montenegro. These national competence centers for HPC in Europe were funded through originally Horizon Europe programs and then through Digital Europe, and all coming from a Euro HPC joint undertaking initiative. This is EuroCC3 now that just started. These were like the largest European projects that are supporting supercomputing competencies in Europe. I think the budget was around 65 million euros, and there is over 35 countries involved. So I'm not promoting only HPC Montenegro here, but I want to have you guys, a lot of you are coming from centers that already established and secured access to high-performance computing resources. So you are doing your research at your universities, companies, etc. But I want to tell you that each and every one of you have an opportunity to go to Euro HPC Center and see what kind of open calls they have. They have open calls throughout the year. So tomorrow, today, if you want to apply, you can apply and you can get access for 3 months or 1 year. And it is open for science and it is open for industry. The only limit for industry is you cannot use it for commercial purposes, but if you are developing new solutions, innovative solutions, you can utilize access to all of the best supercomputing facilities in Europe. So there is, as I said, the slides are a little bit delayed to what I see here and what you see there. Just give me a second. Okay, it takes like 15 seconds. So anyhow, we have over 30 countries from Europe supporting this program. The EuroCC initially was focused on high-performance computing, high-performance data analytics, and artificial intelligence. In the last couple of years, the effort is kind of split the focus. So most of you coming from European countries probably heard about AI factories. So AI factories are a spin-off from the EuroCC. So in Montenegro, Bosnia, maybe Macedonia, Macedonia, maybe not, we still don't have AI factories. So the EuroCC is kind of responsible for artificial intelligence as well. But we expect in Montenegro that we will be initiating some AI factories efforts. So the supercomputing will be one effort, and then AI will be another, and they work, jointly. I believe you are familiar with various supercomputing facilities. Jupiter is the latest one. What we have used here in Montenegro were Lumi in Finland and Leonardo in Italy. So even our University of Donja Gorica, the small university, we had 3 successful calls for 1-year development, access to supercomputing. And again, I encourage you, if you get a need and you can kind of make the application generic enough so that you can use it for different things related to your field of research. And this is a good approach for you to have access to some of the fastest computers in Europe. Besides supporting academia and research, our goal is also to work with companies, with industry, with public sector as well. So for Montenegro, we were very successful. I'll tell you why. When we started this, there were like 0, people using HPC in Montenegro. And even if you ask, IT companies or universities, they were saying, yeah, we don't need that. Everything we do, we can do with regular computing, power. But then you are all witnessing what is happening with this new industrial revolution driven by AI. Everybody is doing some machine learning, model training, video, audio, multimodal. I don't know. There is all kind of crazinesses going on, and all of these activities are requiring supercomputing power or computing resources. So I think we have around 30 or 40 companies already using high performance computing, using our services, and then we are using their services, their experiences, and help to promote the use of supercomputing for mainly machine learning and artificial intelligence application. So some of the use cases involve agriculture, some involve medicine, some involve tourism, cultural heritage, digital marketing, and maybe I'm missing something, but these are all the wonderful applications that I guess you guys are also playing with. So I'm not gonna talk much longer, but I'm just gonna repeat one more time that if some of you need help, you can, ask us here. Stevan is here, I'm here, but you can also Google EuroCC or NCC Competence Center for HPC in your countries. Most of the European countries have one, and you will get an access to a lot of resources on their websites or connect to the people, and they can also help you fill in the applications for the access to European resources. The slides are changing on their own. I was pushing the buttons, so the AI is driving my laptops as I speak. So you can see this wonderful animation going on here. So these are different groups of people that we talk to and that are already using the tools, as you can see. So thank you very much, and I wish you a good continuation of the event and very successful training programs until the end of the event. Thank you very much.
Machine learning service. All right, just a service announcement. I'm gonna read the message because I learned that I'm gonna read this a minute ago. So there's a field trip today. It starts at 14:45. We're gathering in front of the hotel at 3 p.m. We're gonna depart, so there will be no waiting from the buses because the schedule is very tight. We're gonna go to Njegoš Mausoleum and then have a short stop offering a view of the Bay of Kotor. It is the nicest view in the world, probably. Someone from the northern Montenegro will say that there's a better one over there, but one of the best views in the world. It is a steep climb to the Njegoš Mausoleum, so there will be 400 steps to climb. If you don't want to climb the steps, there will be something at the bottom of the stairs for those who don't want to climb to spend a very nice time downstairs. And we're getting back at 18:30. So there will be 1 hour between returning and going for the gala dinner in Podgorica. There's time to shower. There's time to prepare yourself. There's no dress code for the gala dinner, but let's there. It will take place in Podgorica, so it's 20 minutes of driving from here. Transport departs at 19:25, so please be ready to leave 10 minutes earlier in the front of the hotel. The buses also have to leave on time because the schedule is very tight. And you can see, or if you miss the schedule for this, you can see the in the Slack group the number of taxi. So let's say 4 people can pretty cheaply get to Podgorica even by taxi. And we're coming back at 23:15. So we'll be here before midnight. And if anyone wants to return from the gala earlier, there's an emoji that you have to respond with at the Slack group. So be sure to do it as soon as possible because we have to consider separating the buses. So is that all from the PSA for this morning? Thank you.
Thank you, Milutin, and thanks again, Tom, for the nice good morning presentation from the HPC. Our next speaker for today is Maria Brbić. Maria is an assistant professor of computer science and life sciences at EPFL in Switzerland, and she previously completed her postdoc research at Stanford and has a PhD from the University of Zagreb. Her work stands at the intersection of machine learning and biomedicine, with a focus on developing computational methods, discovering patterns in complex single-cell data. And today, her lab builds algorithms designed to generalize to unseen biological contexts, driving discovery in cell mapping and genomics. And through her lecture on AI for biology, she'll explore how machine learning can advance our understanding of fundamental biological systems. And who knows, maybe she also has some interesting response to remarks Phil had on Monday. Well, let's give a round of applause for Maria.
I was an Eastern European student, and now I'm a Eastern European professor. So it's really nice also to have a conference at a place where I can speak the language for a change. So thank you so much for organizing it. It's amazing to be here. So today I will tell you a bit about how AI advances biomedicine and also how biomedicine can advance AI, which is maybe a less obvious direction. So first, we are currently really facing a genomics revolution with the advances of the biotechnology. For example, with advances of single-cell RNA sequencing technologies that I will show you a bit about, we can today measure gene expressions in individual cells. And we can do these measurements in hundreds of thousands or even close to millions of cells in a single experiment. And the status of the technology nowadays is not only that we can measure gene expression, that is transcriptome of a cell, We can also measure proteome, we can measure epigenome, which tells us about epigenome tells us about gene regulation activity. For example, chromatin accessibility tells us which parts of the DNA are accessible for transcription factors and other regulatory proteins to bind, thereby driving the gene expression. It tells us also about the phenome of a cell, that is how cell will look like. For example, reads that you can get from microscopy images and live cell imaging. And also today we can even measure also spatial organization of the cells. So my point here is that today's biology is really nanoscale. That gives us plenty of opportunities. But if we go back like 10 years ago, a bit more than 10 years ago, we could only do bulk sequencing. Why this was problematic is that you could only measure average expressions of the gene or groups of cells. So for example, if you have bulk tumor where you would have tumor cells mixed with the normal cells, you would just get average gene expression. But this is not good enough. So today we have technologies that enable us to measure in each individual cell how much gene is expressed. So now, for example, if you have dissociated tumor, you would take a tumor population, you would dissociate individual cells, you would measure gene expression in each of these individual cells, and this gives you ability actually to resolve different cell populations. Why is it important? Because now you can actually ask, okay, which genes are expressed in these different groups of cells? So it can enable you actually to develop drugs that can target these particular genes and proteins. So this is just very frequent example that people use, very kind of intuitive. Hope no one is hungry, but because you just had breakfast. But before you could only do bulk sequencing, which you can think about as a smoothie. So you take all these kind of fruits together or all these cells together, you mix them, you don't have a clue how many proportions of different cells are there, also what are different kinds of cell types necessarily. While a single cell RNA sequencing is more like a fruit salad where you actually can count on all these individual pieces and understand actually how cells are composed. And not only actually that we can do these measurements, the scale of data is also ever increasing. So when this technology was named Nature Methods of the Year, this was 2013, we could only measure about hundreds of cells in a single experiment. And today we are on a level we can actually measure millions of cells in a single experiment. And this has also driven like large-scale consortia efforts, for example, like a human cell atlas, whose goal is to actually characterize all cells that exist in a human body in health and disease. And not only, again, that we have scale increasing, but also the richness of data is increasing. So as I mentioned earlier, single-cell RNA sequencing just tells us about gene expressions, which is only a part of the cell state. For example, in 2020, the spatially resolved transcriptomics was named the Nature Methods of the Year, which now gives us ability also to know where cells are actually located in a tissue, compared to single-cell RNA sequencing, which is a destructive technology. So you have to dissociate the cell, completely losing the spatial context of a cell. And very recently, in 2024, for example, spatial proteomics has been named Nature Methods of the Year. So nowadays, we increasingly can measure more and more modalities across space. So for example, we can measure, as I mentioned, transcriptome, proteome, spatial organizations of the cells, epigenome, and so on. And with multimodal, multi-omics technologies, this was actually 2019, we can do even multiple measurements at the same time in the same cells. But how, and I don't have to talk about in this community actually also that AI is advancing very rapidly. So we are basically standing at a unique point in history where we have these unprecedented actually abilities of AI systems, but also unprecedented ability to measure biology. And the question is, how can we kind of connect the two? How can AI really help us to drive new biomedical discoveries? How can it help us to advance our understanding of biology and also to understand our cells, how they function, and also to understand what goes wrong on a cellular level in disease state? And our goal ideally here is not just to make predictions, but really to use AI technologies to make new discoveries. And the vision here would be to kind of create this kind of holistic view of a cell state and a human body, that you can measure actually and connect first molecular scale, then the cellular scale, tissue scale, and eventually how all of this forms the complex mechanisms of our own bodies. And as I mentioned, the AI really kind of could be the connection between these different scales and different modalities in order to help us to understand ourselves, model them properly, and eventually engineer cells towards desired cell state, where you can think about how can I go from the disease cell state, like a cancer cell state, how do I need to engineer cells to go back to the healthy cell state. But biology is very complex, because even if we focus only on the cellular scale, as I mentioned, we have so many kind of different layers of regulation: genomics, transcriptomics, epigenomics, phenomics, and how all of this actually eventually affects the phenome, like a morphology of a cell. And we can measure some of these aspects of the cells, but the data is there are still many limitations of the existing technologies. So the hope is that AI could help us to overcome some of these technological limitations. And a holy grail would be really predicting the perturbation response, asking question, what happens if I'm perturb a cell, if I overexpress or underexpress certain gene, what will happen to the cell state? Or if I give a certain drug, how will this affect the cell state? So in this talk, I will mainly focus on our work on multimodality. So in the first part of the talk, which will be a bigger part of the talk, I will talk about how AI advances biomedicine. And in the second part of the talk, I will also show you connections, how actually some of the problems that we are solving in biomedicine also inspire some fundamental AI advances. So first, I will talk about how can we connect some of these different modalities that I have been mentioning earlier. How can we connect the gene expressions of a cell with the phenome of a cell? How can we connect the gene expressions of a cell with the cell organization, with spatial organizations of the cells? And how well can we actually predict this kind of holy grail perturbation response prediction up to date? So we started this project with the idea and the goal in mind to link the transcriptome and the nuclear morphology. So in particular, we were interested to understand how genes or kind of transcriptional programs give rise to the nuclear morphology, and how also morphology influences the cell state. And there have been prior works studying these connections, but the unique aspect here is that for the first time, we actually had the paired measurements that give us a full transcriptomic profile of a cell and the nuclear morphology of a cell. So I think in this community that many people, I guess, are thinking about multimodality, I don't have to say how precious actually this data is, where we are measuring things at the same time in the same cells, which really gives us true multimodal data. However, in this case, as I will say later, we don't have plenty of these multimodal measurements. So these measurements in particular, they come from Par de Planque's lab, where they just developed a new technology that they call IRIS. And using IRIS, cells are loaded in microfluidic chip. They're precisely placed and imaged, and then encapsulated in a droplet to label cell mRNA molecules. So this basically gives you an image of a cell and then full gene expression profile. And here, when I say full, I'm talking about around 20,000 genes. And also high resolution image as well. So using this data and led by my PhD student, Shuo, we were thinking about how can we actually now develop a model that can help us to first link these two modalities, to quantify how well can we go from one modality to another, how well can we predict one from another, to quantify this relationship and the link. And then also, ideally, if we can quantify to a good extent, can we actually learn about underlying biology? Can we learn which gene expressions are associated with nuclear morphological features? And this is actually very frequently the case when you're developing models for biology, that you also don't want to have only predictive models, but also gain some insights why things are happening the way they are happening. So as I mentioned earlier, so what we want to do is we conditioned on the microscope image of a cell. We want to be able to see how well can we predict the gene expressions of a cell. So this is a vector, about 20,000 dimensional vector. And then also how well, given a transcriptome of a cell, can we predict the image. So here we don't have actually plenty, as I mentioned, like multimodal measurements. You cannot just go and just take this pair data and just hope it will work, because here we have about 10,000 samples, which is in machine learning I think a few thousand, actually, less than 10,000, which is actually in machine learning sense pretty small. So our approach was, can we first develop like a strong encoders of each of these modalities, and then learn how to link them even with less multimodal data. So in particular, for the transcriptome of a cell, if you think about how can we encode the gene expressions of a cell, there have been plenty of the methods already developed. So who is actually familiar with like generally modeling single cell data in the audience? So 4 students, 2 from my lab, okay? Sounds good. So we have different encoders that you can use that already exist. These are kind of so-called foundation models for single cell, like universal cell embeddings, or like single cell GPT, which, as the name says, it's kind of a GPT-like model for biology. But there are also some smaller models like, for example, SCVI, which is just basically a variational inference model, which is just simple autoencoder that you can train on the data without any kind of large-scale pre-training. But we also needed a strong nuclear morphology encoder, so we decided to build our own. And this was also supported by the Swiss AI, which is actually a huge initiative in Switzerland, where we have access to about 10,000 H100 GPUs for this kind of large-scale pre-training of models, which gives us really ability to think about ambitious projects and also like this large-scale training. So what we do, there is a good news that we have lots of nuclear morphology images that are publicly available. So what we did, we went widely and just like collect the data on our own. So we train this model on over 22 million nuclear microscopy images. In particular, you start with the whole well microscopy images, then we segmented out them to get like individual nuclei. And then we train this model in the masked autoencoder fashion, pre-train the model. So basically, you mask the parts of the input patches, and then you train encoder and decoder to predict those match patches that have been masked. So this is standard mask autoencoder pre-training. So this we trained basically vision transformer large with about 300 million parameters. Then conditioned on these conditioned on the representations from the encoder that we learn, now we want to basically learn the link between the 2 different modalities, for which we use the paired iris data that I mentioned earlier. So in particular, we train conditional diffusion models where conditioned on the transcriptome of a cell, we generate the morphology, and vice versa, conditioned on the morphology, we generate the transcriptome of a cell. Yeah, so this is exactly the statistics of the data. So for human cells, we had about 9,000 samples, paired samples. For mouse data, we had more, like 70,000, but we trained them separately because genes are different across different species. So first we go from the transcriptome to the nuclear morphology. I will show you some results. So the question is, of course, also how to evaluate these generative models. Because what you get, you conditioned in this case, you conditioned on the transcriptome, you generate the image, but the question is how well does your image reflect the ground truth image? So first we embedded these images using this large model that we pre-trained in this 2-dimensional UMAP space. And kind of generally, it seems here cells are colored based on different cell types. So you can see that the model was able to model correctly actually these differences between the different cell types. And if you look at some examples, visually, at least to us as humans, it looks like cells from different cell types look similar to each other. But now if you want to quantify this and compare to existing methods, for example, we adopt very different metrics, but here I show an example of Wasserstein distance. You can see actually that cosmic space actually resembles the ground truth space compared to some other methods that you can apply for this task, where, as you can see, the distance is much larger, but also the underlying embedding space is much worse too. So now this is the comparison to baselines. Continue to share. Okay. Now, this is just a comparison of the baselines in the image space. Here you can see actually that existing baselines are not able to correctly model actually how cells look like, also even visually to us as humans. And another thing what we do to evaluate is that we train the classifier on the ground truth images to distinguish between different cell types. And then on this, we freeze this classifier. Just during inference, we input these kind of different generated images generated using different models. And see how well this classifier trained on the ground truth images can now separate between these different cell types in the generated images. And you can see actually for cosmic, this performance almost approaches the performance, the upper bound you would get if you train it on the ground truth images, while for other baselines, it's much less. Next, I will show you the other direction, which arguably is also much more interesting direction. Why? Because sequencing data is very expensive, while imaging is very cheap. If from the image you can kind of directly predict expressions of some genes, this would have a huge value and clinical implications because it could be directly adopted, for example, in clinics, where you could potentially be able to predict expressions of some genes directly from the image. So now this is actually the other direction. So some examples, again, now this is the gene expression space. We're conditioned on the image we are generating gene expressions. So here, these yellow dots are cells that are predicted by cosmic, and the blue are ground truth cells, where you can see actually large agreement between the two. And also, even if you look beyond the cell type information, which is quite coarse, how well, for example, the model is modeling the cell cycle, which is dynamic process. You can see actually that the expressions of the cell cycle genes between cosmic and real images are agreeing decently well. Again, a comparison to different baselines shows that cosmic is much better, but more interestingly, there are more than 15% of genes that have correlation higher than 0.4. So next, we were interesting, actually, can we now identify those genes? So given that the model is kind of predictable in both directions, can it tell us something about genes that are associated with the nuclear morphology? So to do so, we first use cosmic sequence to image to generate from the transcriptome the image itself, then image to sequence part of the cosmic to generate the transcriptome back to the transcriptome. And then we calculate the percent correlation for each genes between the starting point and the end point. And here, key idea is that only nuclear morphology-related genes can pass this information flow. So we use this approach to identify those genes that are associated with nuclear morphology. So this is not causal. For example, for causal relationship, you would need to have perturbational data. So this just tells you about the potential associations. So now this is an example of what it tells you. So it tells you basically you can find those genes who, if they have a low expression, the cells look very differently, while if they have high expressions, the cells are changing actually their shape. So this is just an example of one of the high rank genes, where you can see actually with the high expressions, the cells are much bigger, while with low expression cells look much smaller. Now you can also run some kind of cell profiler, which gives you actually some particular morphological features of the cells that you can study. For example, the mean intensity between these two cells is very different, as well as the variance. So now using this approach, we were able to identify some genes, actually quite interesting genes. So this is just an example of one such gene where, for example, we find these genes with previously unknown function, where we can see that actually independently across all these different immune cells, it has differences in the morphological features, depending whether the gene is highly or lowly expressed. So this could kind of have a way to study some genes that for which functions or morphology associations were previously unknown. You can use, for example, cosmic to tell you about such associations. And finally, we really like also in the lab collaborating with biology. So we collaborated with Walter Karthaus' lab at EPFL, where they're interested in the prostate cancer cells. So what we did is we took a group of prostate cancer cells. One were non-treated cells, and then the other cells, these were control cells. Other cells were given a drug treatment, in particular Taxol treatment. So what we've seen then is that some groups of cells that received the treatment were grouping with these non-treated cells in this cosmic embedding space, while other cells were forming their own cluster. Now, inspecting using this kind of cosmic to find those genes associated with these changes, we were able to identify some genes that actually correlate with these differences. And some of these genes are genes that are known to be associated, for example, with cancer, like generally with cancer aggressiveness and progression. So again, this could kind of give you a mean to study actually these associations between the morphology and the transcriptome of a cell. So next, I would like to talk about how we can go from cells to tissues. So in particular, if you start with dissociated single cells, we ask the question, how can you predict a tissue structure? So on a high level, you can think, I guess many of you have heard of AlphaFold, which predicts the protein structure. Here, the idea is more kind of AlphaFold for cells, where you start with dissociated cells, where you don't have information about their locations, and you want to predict these kind of complex tissue structures and how cells get organized in the space. So we call this process tissue reassembly because we start with this, we try to reassemble cells into meaningful tissue structures. So why is it important? There is a practical importance in a sense that single cell RNA sequencing technologies are destructive, which means that you completely lose the spatial context of the cells. While we actually care where cells are located to study, generally to study, for example, tumor microenvironments, disease, but also in the healthy cells as well. And secondly, the reason why it is important because it could inform us actually how cells are forming tissue structures for potentially some underlying rules in how cells are organizing, like underlying mechanisms of these kind of complex biological processes. But the question is also to what extent is tissue reassembly even possible, given vast cellular heterogeneity, complexity of tissue structures, intricate cell-cell interactions, and also elusive biological mechanisms that underlie tissue formation. And if it is indeed possible, how can we learn it? How can we learn these underlying rules of complex spatial organization? So what we did is we took, good news is that there are lots of existing spatial transcriptomics data sets. So spatial transcriptomics technologies that I mentioned briefly before enable you to measure cells in their native tissue context. So they're not destructive, but unfortunately you cannot measure full gene expression space. You typically can measure about 1,000 genes at most in a single experiment. So the idea here was that we leverage these existing spatial transcriptomics data sets to learn underlying rules. And then we would be able ideally to apply these rules to the single cell RNA sequencing data. So the challenge is here that we need to capture cell interactions locally and globally across the entire tissue slice. And with this goal in mind, we developed Luna, where this was a work developed by my PhD student Yeast and master student Shanakia. Of course, we've got also contributions with other people, but they were the key people developing the framework. So there are two key ideas in Luna. So first, we use the attention mechanism in order for each cell to learn how much importance should it give to other cells in the context. So every cell needs to learn how it should attend to other cells based on the relevance of other cells to the cell's all locations and to the gene expressions. And the second thing is that we model this tissue reassembly process as a generative process. So in particular, using conditional diffusion models, we want to start with the noisy cell coordinates. Then we want to condition these noisy cell coordinates on the gene expressions of the cells. And then with a number of diffusion steps, ideally be able to reconstruct the tissue structures. So next, I will explain how Luna framework works. In particular, as I mentioned, Luna leverages spatial transcriptomics data with ground truth locations. So you start with the ground truth locations, and then you sample diffusion time. So as a diffusion process, you have two main steps, corruption and denoising. So during corruption process, we are adding noise to the ground truth cell locations. And then during the denoising process, we are learning to recover original cell coordinates conditioned on the gene expressions of the cells. So basically, we sample diffusion time, then you go to, from the ground truth cell coordinates, you go to the noisy cell coordinates. Then you learn the cell embeddings using multi-head self-attention, where every cell needs to learn how much importance should it give to other cells in the context. Then we add another MLP layer to predict when final cell coordinates and introduce, and then you want to predict basically your objective function is to predict the clean cell coordinates from these cell coordinates that were corrupted by noise. And we introduce rotation invariant loss, so we don't directly predict the cell coordinates. We actually predict the pairwise relationships between the cells because spatial arrangements of the cells may undergo transformations due to experimental artifacts. So basically cell slices could be arbitrarily rotated and translated. But you want actually, but the gene expression profiles would remain unchanged. So basically the model has to be invariant to these arbitrary rotations and the translations of the input slice. So this means that the objective has to be invariant to these arbitrary rotations and translations. So we use the pairwise loss where we basically focus on the pairwise relative distances between the cells in the slice. And during inference, we sample from standard normal distribution, and then with a number of diffusion steps, ideally, we would like to be able to predict the tissue structure. So Luna has a time and memory complexity that is linear during inference. During training, it's not linear because you have this pairwise loss. But during inference, it can infer locations of tens of thousands of cells in just a few minutes. And we use the basically attention with linear complexity and show that using the approximation, you get similar performance as if you compute the full attention. So now next I will show you some exciting results. So first we applied Luna to reassemble mouse brain. So in particular, we train Luna on about 2.8 million cells from one mouse, and then we applied to about 1.2 million cells from another mouse. Now these are ground truth cell locations. These are Luna's predictions. Cells, each individual dot represents a cell. Cells are colored based on different cell types. And here with the number of diffusion steps, you can see actually how diffusion process happens. And with the number of diffusion steps, model converges to the ground truth locations. So generally, we see good performance across different slices. You can see actually and appreciate how differently these slices really look like because it's an entire mouse brain profiled. But across 11 major regions that are identified in this atlas, despite their structural characteristics, we see the model is actually able to model them well. Next, we were asked, actually interesting to see whether model can generalize in the zero-shot fashion. Given the high similarity of these two mouse brains, we were interested in what would happen if we exclude completely one of the major cell types one by one to see actually how well can model generalize to different cell types that it has never seen. Can it actually correctly place them with the spatial architecture of the tissue? So now basically we completely remove certain cell classes and see whether model, even though it has never seen them during training, can also correctly predict them in another mouse. Now this is an example for glutamatergic neuronal cells where you can actually see that the model was able to pretty correctly actually position where these cells would look like, where these cells would be positioned. And also if you look at some genes that are changing expressions across the space, for example, these are known as spatially variable genes, you can actually see that the model is able to correctly place these genes that are expressed highly in this area region are actually positioned here also by the model. Cells that are expressing those genes, and similar for another spatially variable genes. So this is also the case for other unseen classes, which have also very different distribution. For example, vascular cell class, you can see very different distribution than in the previous slide. But again, model was able to correctly place cells and genes that are expressing those cells. So Luna is a unique method in this idea that we leverage actually multiple slices to then generalize. But if you look at existing baselines, there are two groups of existing baselines. So one group of baselines are methods that map dissociated cells to a reference slice, but you have to have exact reference one from another. So you cannot actually learn these kind of more generalizable transferability. You have to have exactly same slice from the single cell RNA sequencing data to the spatial data, which is actually quite limited in practice. And the second group of methods are methods that are kind of trying to do the similar thing as Luna, but they, instead of learning these spatial priors, they put assumptions. So they say if they solve the optimal transport problem by saying if cells have cells that have similar gene expressions should be spatially close together. But this actually assumption is not correct for very complex tissues. So that's why these models fail in these scenarios that we test them, as you will see. So here now this is comparison to Luna and to other baselines, where Luna actually we measure whether the model was correctly using Spearman rank correlation compared to the ground truth cell locations, whether this kind of neighborhood similarity is correctly rank ordering of the neighborhood similarity is correct. And you can see actually that other methods severely fail compared to Luna. And even if we give some priors to existing methods, so we actually tell them and give them exact reference slice, so we help them and use the information that Luna is never using, the model is still much better than existing baselines. So next we use the model really to go wild and predict from the dissociated cells. So all these previous results were actually where we could validate whether model is correctly placing cells. But our ultimate goal is really be able to make predictions for another technology, single cell RNA sequencing, where also distribution shifts when you try to transfer, where we don't have any information about the locations. So now we use the model that was pre-trained in the same way that I introduced before, but now during inference, we actually input the single cell RNA sequencing data where we don't have the spatial coordinates. So we estimate the locations by mapping those cells using another technology called StarMap that the model has actually never seen. This is just used as a validation whether approximately these two approaches agree. So one is actually using the technology. You try to measure the spatial organization of the cells and then map single cell RNA sequencing to the spatial. And here, actually, these are Luna's predictions. So now what you can see is actually that Luna's predictions agree pretty decently with this kind of estimated ground truth locations. And also, if you look at the expressions of some interesting spatially variable genes, like, for example, IQGAP2 genes, which shows a gradual downregulation across the Ammons home region, you can see actually that these kind of expressions are well captured by the model. Why is this interesting? Because now, because the Luna was trained actually on much smaller number of genes. Now what you can do is now you can actually take these 20,000 genes for which you didn't have a clue where the cells expressing these genes could be. Now you can actually map them to the space and identify existing or like new previously unknown spatially variable genes. Next, we were looking also to kind of get a bit of insights what is happening in the model. So in particular, during training, we used all genes to train the Luna. Then we removed gene by one, kind of simulating in silico gene knockout. And then we were interested to measure how much actually removing gene would affect the spatial predictions of Luna in order to identify those genes that are kind of contributing the most to the model's predictions. This kind of gives us insights actually into genes that are important for certain cell types, which actually would then tell us which these genes are indeed genes that are crucial for cell identity and function, but they were not previously associated with kind of their effects on the spatial architecture of the tissue. And then finally, we collaborated with Joelle Lamano's lab at EPFL. So in Joelle's lab, they performed experiments. They injected alpha-synuclein, which is a protein associated with the Parkinson's disease. They injected it into mouse brain and then collected brains 3 months post-injection. And next, we were interested to see, actually, can we now be able to train the model on the healthy samples, identify potentially those regions in the space without ever seeing the spatial locations that are identified, that are affected by the pathology of by disease pathology. So in particular, we trained the Luna on the healthy samples, but now during inference, we apply Luna to the Parkinson's disease model that model has never seen. we aligned to the common coordinate framework using Luna healthy and disease samples in order to identify potentially regions in the space that are changed by the disease. Then we performed immunostaining for alpha-synuclein to actually identify where this protein is expressed. And indeed, one of those regions that was actually predicted by the model is the region that is actually expressing this alpha-synuclein. The next, like my last vignette about biology is thinking about actually, are we done and foundation models are the ultimate solution. So far, as I mentioned, there have been different foundation models developed also for single cell biology, also for other biological modalities. And in particular, the task that we care a lot is kind of perturbation response prediction. So there the idea is that you take a bunch of these single cell data sets across different experiments, you train them on the lots of data, typically masked by masking particular genes and then predicting the expressions of the genes that have been masked. And then you apply these models to a variety of downstream tasks that we would expect these models to generalize to. And one of the most important tasks is this perturbation response prediction task, as I will talk about. And this work has been driven by my former postdoc, Ramon, who is now at Isomorphic Labs. So in particular, we asked the question, how well can we predict response to unseen genetic perturbations? So what would happen, so what this means is that you have a group of control cells, you apply some perturbations, so this means you express, you underexpress or knock out certain gene or overexpress certain gene. And now you expect, you would like to predict what would happen to the gene expressions after. So what would be this post-perturbation profile given the perturbation that has been applied and the expressions of the control cells? And there have been a number of methods in the literature, including these foundation models that have been claiming that they can predict the response to unseen genetic perturbations. So in particular, you have a set of trained perturbations. Then you have some test perturbations that you have never seen during model training, and you would like to predict how cells will respond to the unseen genetic perturbations. How do these models work? So there are two groups of methods. So one would be these kind of foundation models like single cell GPT, where what you would do, as I mentioned, you would train on lots of the data, mask expressions of certain genes, try to predict the expressions of those genes. And the assumption here is that although you have never seen these genes being perturbed, kind of the model was able to see these genes during pre-training, so it kind of has learned these kind of correlations between different genes. There are also some specialized models that kind of have some prior knowledge. For example, one example of such methods would be like gears, for example, that they use gene ontology, like which genes have more similar functions, and use this kind of information to investigate, to have some proper priors to generalize to unseen genetic perturbations. So what we did is we collected about 10 different perturbation data sets across 3 different technologies, 5 different cell lines. This was also collaboration with Sarah Teichmann and Mornitsan. And we asked the question to what extent can we predict these transcriptional outcomes of unseen genetic perturbations. So what is the metric that people use is this kind of Pearson correlation between predicted and ground truth expression changes. So before and after perturbation, you measure the Pearson correlation with respect to the control cells. You can measure this on all genes or, for example, top 20 most impacted genes. And we construct a very simple baseline, which we call perturbed mean, where you just take the average expressions of all perturbed cells and use this average as your prediction for all unseen genetic perturbations. So you basically never trained any model, you just do the averaging. So surprisingly, we show that this kind of super simple baselines outperforms or performs as good as these existing state-of-the-art methods. And this is also the case for combinatorial perturbations, where again, there are like very simple baselines. I won't explain in the details, but just some averaging, no model training, anything. And then again, we see that this simple baseline on this most frequently benchmarked data set Norman is actually better than these huge foundation models. So the question is, why do simple baselines? So how am I with time, Peter? Okay. Okay, sounds good. So the question is, why do simple baselines perform so well? So we assume that there is kind of some systematic differences between control and perturbed cells. So these systematic differences could be caused, for example, by the way genes to be perturbed are selected, or for example, by underlying biology. So I'll show you just some example because we investigate this across all these different perturbation data sets. But you can see, for example, in this data set, if you measure cell cycle distribution between control and perturbed cells, there are actually significant differences across different cell cycle phases between control and perturbed cells, which can be explained by the fact that p53 positive RPR1 cells with abnormal karyotypes tend to react to chromosomal instabilities that are caused by perturbations through cell cycle arrest. So that's why you see these kind of systematic changes between control and perturbed cells. So the assumption here is actually that existing methods are just measuring and capturing these kind of systematic differences between control and perturbed cells, rather than really being able to predict perturbation-specific effects. And now we went, as computational people, we went to quantify this degree of systematic variation. You can measure, you can start with the control cells. You can have a vector that goes from the control cells to the perturbed cells. So this is just measuring the average perturbation shift. And perturbation-specific effects would be vectors that go from the control cells to the centroid of cells that underwent the same type of perturbation. Now you can measure cosine similarities, and what you can see that, and this is how we measure actually the systematic differences of systematic variation. So basically high cosine similarity indicates that all shifts are pointing in a similar direction, which suggests shared, possibly non-specific shifts in gene expression. Now you can see that actually data sets that are very frequently benchmarked have this high degree of systematic variation. And if we now correlate this degree of systematic variation with the performance of these methods, actually there is a high correlation between the two. So this means the more systematic variation, you would expect higher performance, which shows that commonly used metrics that we are using actually to measure how well these models are performing are inflated by these systematic performance and systematic differences. Now this motivated us to propose Systema, which is a evaluation framework for perturbation response prediction that accounts for this and asks, okay, how can we really well predict this perturbation specific effects, accounting for these systematic differences between the two groups? And what we show is actually that we change the reference from the control of the perturbed cells, from the control of the centroid of control cells to the centroid of perturbed cells to emphasize these perturbation-specific effects. And interestingly now, when we do that, you don't see this kind of correlation anymore with systematic differences and the Pearson correlation. But more interestingly is that now, actually, if you see how well existing methods predict these perturbation-specific effects, the correlations are no longer like 0.8 or 7. They are actually very, very low, which actually shows that existing methods can still not well predict these responses to the unseen genetic perturbations. And this actually shows how important is the metrics and how important is our evaluation. How are we evaluating these models in biology? Are we using proper metrics? Are we accounting for the proper biases that could happen in this data? And in the last part of my talk, I would also give you like two examples. And in this talk, I've focused mainly, although we also work more broadly on different problems. For example, Siba, who is here, works on agentic models as well. I will focus here more on the multimodality. So I will give you kind of two vignettes of also kind of how some of the works that we're doing in the lab in biomedicine can also inspire some like AI advances that we apply more generally beyond biology. So first, as I mentioned, while we were working on a cosmic, there was this problem of that we were training. Actually, we had a small amount of the paired data sets. But typically, when you think about how we train these large vision language models, they're trained on hundreds of millions of the paired samples. However, in biology, in biomedicine, medicine in general, millions of paired samples are often unavailable. We just cannot connect them. We cannot collect them because these are coming from experimental We have experimental limitations, as I introduced earlier, or for example, in medicine, when you try to collect information about patients, very often you cannot have like millions of like paired modalities that come from patients. So the idea here is that, what would be approach if we don't have lots of multimodal data? Instead of training from scratch, where you would take this multimodal data, train multimodal model, what you can try to do is you can try to align pre-trained models. So you can train the model on lots of unimodal data. For example, this would be like vision language, or could be just like histopathology images, gene expressions, for example. And then you can try to build the alignment between these two. And this has been, of course, also adopted in the machine learning literature. So you can just take, your two modalities, you can freeze their encoders, and then you can train alignment function. Typically, this would be contrastive objective that tells that samples that are similar to each other should be close also in the embedding space, and samples that are different should be far away from each other. But in this existing literature, these kind of models still require millions of paired samples. And our question was, can we use less than 1% of the data that is typically used in the field? And this work has been led by my visiting PhD student, Fabian, who will now join us as a postdoc, and my PhD student, Shuo. So in particular, what we did here is that the idea here is that in this case, when you don't have many multimodal measurements, maybe there's some way to introduce some regularization that could help you to do this more effectively. And here what we do is that we measure the similarity of points in the pre-trained space. And the idea here is that we want to preserve the structure that we have in the pre-trained space after we train for the alignment. So in particular, the key idea is that you want data points that are similar to each other in the pre-trained space to remain similar in the aligned space. So you can start with your pre-trained space. You can measure similarity matrices. Basically, you interpret these similarities as probability distribution. You can just apply a softmax row-wise after. And then what you can do, so this you just transform the similarity distributions, and then what you can do is you can measure Jensen-Shannon divergence between these two distributions. And you can minimize this actually distribution before and after pre-training. So basically in the shared space and also in your pre-trained space. And now we add this regularization. So this is the resulting regularization. We can kind of capture these relationships reachable by exactly L hops on the similarity graph. So we use L, we show actually that with the larger L, you don't get much boost in performance. So we set L to the first order neighborhood. And now you can add this regularization to any alignment function. So you can add this regularization for your modality 1 or, for example, for modality 2. And this is kind of super simple. You can incorporate it and add it to any kind of alignment function. It's very simple and like modular framework. So what we show is now if you want to do linear alignment with this regularization, you achieve about 68% average improvement in this kind of scenarios where you have small amount of the multimodal data. So here we train these models, for example, using only 80,000 samples, which is actually less than 1% of data used in the field. And we see this improvement across both zero-shot classification and retrieval tasks. Similar for MLP alignment, for nonlinear alignment, you achieve similar, even larger actually improvement in this scenario. So even if you have less than the 80K samples, so even if you scale, you can see actually that the structure helps. So this is with the added regularization. This is training without the regularization, the blue curve. So again, we see this both for zero-shot classification and retrieval tasks. So despite these advantages of this kind of alignment approach, you can notice that there are actually some tasks, I highlighted them here, for which just the overall performance is still quite low for the multimodal alignment. But interestingly, if you just add a few labeled examples from the target domain, you can see that the model, so this is that the models can bridge the performance gap to large-scale multimodal models that are trained on hundreds of millions of paired samples. And here actually is the performance of the CLIP model, for example, that you would train. Okay, and in the last part of my talk, yeah, I would like to discuss kind of this revisiting the Platonic representation hypothesis. I think you heard about it from Philip Isola in the first part of the talk. In the first part of the week, I think Philip was on Monday, I think. I would like to kind of give maybe just a slightly refined picture of this hypothesis. And this work, again, has been driven by really fantastic students, Fabian and Shuo. So you saw Shuo also on many slides today. I think you're now familiar with the Platonic representation hypothesis, but I will state. So this was presented by Philip and his collaborators at ICML 2024. Which says that basically neural network trained with different objectives on different data and modalities are converging to a shared statistical model of reality in their representation spaces. So what this means is that they measure the distance between the data points in more and more alike way. And there was lots of discussion in the community after this was presented. Is this really the case? Some people were really, fascinated and so excited that actually the, vision and the language models independently trained are kind of converging to the same representation space, which is really exciting thought. And on the other hand, some people were skeptical, this may not be the case, and so on. But no one actually has shown that this is not the case. So what is the Platonic evidence? So this is from Philip's paper. If you, so you have a language model, and you measure the similarity, representation similarity to, for example, a language model, and you can use different metrics. So they show it robustly across different metrics, like this is a center kernel alignment. They also show CCA, canonical correlation analysis, also mutual care, nearest neighbors, and so on. You can see actually that as the models are growing larger, the way that they represent the way they represent the world kind of becomes more similar. So the representations of these models are becoming more and more similar to each other. So in general, like bigger models show better alignment. And we saw this also across different combinations of the different language models. So here on the x-axis are different language models. And this is now alignment to Dino, but you can see actually this trend being actually similar when you change the vision model. So this is now CLIP and MAE and so on. So the question is, how are we measuring the alignment? So how you measure alignment is that you compute representation similarity for each layer combination. And you, because you have many layers, so these networks have many different layers, so you would compute the representation similarity pairwise across different layers, take the maximum value to report. And again, you can use different metrics. You can measure CCA, CKA, and so on. But then eventually you just aggregate across all these comparisons. So we asked the question, what if you're not measuring this representation similarity properly? So in particular, we show that existing representation similarity metrics are confounded by network scale, in particular network depth and network width. So there is a width effect where global representation similarity metrics, and by global, these are, for example, CCA and CKA, these similarities are growing as D over N. So we call this width effect. So what this means is that the wider models have higher spurious global representational similarity. And the depth effect says that basically that maximum similarity grows as L. And I will explain why this happens. So this means that deeper models have higher spurious similarity. So if you look at the width effect, so now even if you have two independent vectors, you measure CKA and CCA across different N and different D, basically these similarities are growing as we increase d with respect when you increase the d relative to increasing the n. As a result, wider models may appear more aligned simply because their representations live in higher dimensional spaces. So basically, there is kind of this positive, systematic positive bias when measuring this, even when representations are independent. And what is the depth effect? Depth effect happens because just from like statistics, basically, the more comparison you do, so as I said, you take the pairwise comparison, you take the max value. But the more comparison you do, you're just more likely to find an effect just from the statistical perspective. So basically, the more tickets you buy, the higher the chance you win a lottery. So now we basically adjust for these metrics. So we calibrate for these actually systematic biases. So what we do is you can just construct empirical null distribution by breaking the correspondences between vision and language pairs. And you construct the null distribution when there is no actual relationship between vision and language models. And then you measure how extreme the value graph is that you're actually observing compared to this null distribution. And this kind of gives you a calibrated score. And now we ask the question, okay, once we calibrate, and we can apply this calibration to any metrics. So you can apply to CK, CCA, mutual KNN. So it's independent of the metric. So then we ask the question, okay, so what happens to the Platonic representation hypothesis after we calibrate? Now what we showed that is so this was before the calibration. So these are results reported in the original paper. Now if you measure global similarity, for example, CKA, but similar for other global similarity metrics, you can actually see now these are actually language models. This is alignment to Dino, and this is alignment to CLIP. You can see actually that it's no longer the case that as we increase the scale of these models, that the similarity is increasing. So basically, after calibration, the trend of global alignment disappears. If you look at the local similarity metrics, for example, mutual KNN, we still see this effect. And this is also because mutual KNN, it doesn't depend on the dimension. So it doesn't depend on the D. It's just, you just measure the local neighborhood relationships. So although it's sensitive to this L effect, to the depth effect, it's not sensitive to the width effect. So after calibration, the trend of local alignment actually still remains strong. And I think this is kind of another example where actually measuring things properly is important, like similar what I made for this perturbation response prediction task. And this motivated us to propose an alternative representation, Aristotelian representation hypothesis, which tells us actually that neural networks trained with different objectives on different data modalities, what we can only claim based on empirical evidence is they are converging to the local neighborhood, the same local neighborhood relationships, not even local neighborhood distances, but just local neighborhood relationships. This also recently motivated us, so we will soon put this paper on archive to actually, that you can have some way to kind of, before aligning the models, so this is just a spoiler, but you can take like different pairs of the models, like vision language models, and before even aligning them by computing some of these metrics, in particular, two scores that we call ease and ceiling, which are actually based on the theory of this, what I introduced before, a mutual K nearest neighbors. You can actually compute before alignment how easy it is to align specific pairs of vision language models. And this can give you pairs on which you can kind of act now, because you expect that after pre-training, you can they will have a high alignment score. And we actually show that this pre-transfer diagnostics highly correlates with what you expect, what you get after you actually align the model. So this is kind of a way to effectively tell you before aligning, before expensive training, which pairs should you focus on. And to decide if two models are compatible or for alignment prior to training. So finally, I would like to thank first like my amazing students. So the students whose work I presented today are highlighted in boldface. They are the one really doing amazing work. And also our great collaborators who make all this science fun. We are also hiring both PhD students and also postdocs. So if you're interested, you can find me after. Happy to chat. Thank you so much, and I'm happy to take questions.
All right. Thank you so much, Maria, for the very engaging and very exciting talk. We have time for 3 or 4 questions from the audience. Any questions?
Hi.
Hi.
Do you think that the way on how we represent as humans things like thoughts or so is also a way on how we can measure the differences between how neural networks by themselves also represent them, like in the sense that I think in medicine, I mean, no one really has the same diagnosis or the same process in that sense. It's also a way to measure the similarity, or it's completely different?
You mean in medical applications?
Yeah.
I mean, it's hard to kind of quantify, right, how one measures thought in generally, right? What you can measure potentially is, I mean, I think in medicine, it's, yeah, I think you would need, it's mainly so far based, I would say, on some post hoc, right, analysis where you have some, I know, diagnostics given from one person, like one doctor, medical doctor, another medical doctor, there is a disagreement. Now you can measure after the disagreement to the human, but it's kind of difficult to measure representations, like latent representations of a human brain compared to you know, because this is now this is done like what last part is done in like representation space, right? But in a representation space, it's hard to measure our own representations. We can only measure post hoc what happens, right? These are the predictions of the expert versus predictions of the model. Yeah, maybe some neuroscience, you know, like related work can come with some, you know, approaches for something like that. Yeah.
Thank you. Any other questions?
Hi, I have a question about the first research paper or the first part about using AI for medicine. Is there a specific area or thing like in the paper that needed more research or was under researched that needed more details or work to be done on?
You mean generally, are there some underexplored areas in
Yeah.
AI for biomedicine? Yes, I think so. I think most people so far are kind of, many people are focusing, I think, on understanding, for example, gene expressions, right? Because this is the modality where we have like lots of data. But proteins, which actually, but eventually we care about protein products, right? So we care about proteins really. So, but I think in the protein space, there's kind of this proteomics technology, like a spatial proteomics and so on. I think there's lots of actually existing data sets there, but maybe there's less research compared to the transcriptomics community. So I would say this is definitely something. And I think another thing I would say is maybe also thinking about, you know, connecting some different aspects together, for example, like live cell imaging, which is actually gives you information about, you know, what is happening cells temporally, right? Somehow thinking about how one could connect this with these kind of technologies that are destructive like single cell data, because so that you can kind of say something about time and evolution, like development of tissues or cells across space, like this kind of spatial temporal dynamics, which would be very interesting and is crucial for biology to study. But I would say it's not as mainstream as this kind of, let's just take these kind of observational data sets or perturbational measurements and try to do things with that.
Thank you.
Thank you.
Thank you. Any other questions for Maria? Okay, I would like to ask one question. I think this was brought up a few times during the talk, but I'm just curious because in some parts of biological AI research, we're starting to see a trend to like reduce constraints on the model, reduce the explicit use of symmetries and these kinds of things. But then a lot of like, rare disease data and things like that tend to be very data, constrained. So I'm curious, what are your thoughts on like the future of the use of symmetry in this space?
I think these are useful priors. And I think whenever we work with biological data, we really have to think about, what is the proper, because I think very often what we see so far in the community, just this approach, let's fit more data and hope it works, it just doesn't work. Right. So in a sense, I think having priors that actually account for like what the data looks like, I think still are crucial in biology. And I think we still don't have like really good data sets to train like really foundation models. I think these are just like large scale models that are learning somewhat useful representations, but they're not foundational in a sense that large language models are foundational models. So I think there is still really thinking a lot also about also kind of how these data symmetries could help us maybe also for the data like experimental design and like what kind of data sets to generate, even like driving new experiments potentially. I think there are lots of opportunities.
Thank you for that. All right, let's thank Maria one more time. And we will be transitioning straight into the UiPath keynote, after which we will have our first coffee break. I would invite the UiPath speaker to please join us here. Yes, hello. And Matko, would you like to step up as well? Thank you.
Hello.
That's a mess. Fancy picture.
So we're back again. Our next speaker, our next industry keynote speaker is, I'm sorry if I'm going to somehow butcher your last name, Stanuts.
Yeah, it's very good.
So Nikolai Radu Stanuts, who's representing our school's sponsor, UiPath. So Radu is a senior applied scientist at UiPath, where he focuses on advanced automation architectures and computer vision driven agents. And as you can see, like his work centers on bridging traditional programmatic workflows with computer use agents. And he's going to talk about computer use agents from scripted bots to code workers. I'm kind of happy to see people working on this because I'd like to have more able agents to do more of my job so I can go more to the beach.
Yeah, actually, they did my presentation, so.
So yeah, we're very grateful for UiPath supports. We're very grateful for Radu's talk, and Radu, give it away.
Thank you for having me. So I know I'm the only thing standing between you and the coffee, so I'll be quick. So please meet Otto. Otto's been with us since 2005. He's been helping our clients to automate their work. Of course, Otto is the mascot here, the helicopter guy. I wish I were with UiPath since 2005. If you don't know, ask a Romanian. They are on yachts right now. We are Otto's colleagues, Maria, Stefan, and me. And we, as I said,
we automate the boring stuff. So things like Laura's afternoon. Imagine Laura's afternoon. Laura is a back office worker. She receives a dispute on an invoice. She has to open that invoice, read manually the amount, and enter it in who knows what ERP. I don't know if you are familiar with this kind of work, but it may seem quick, but it's repetitive and it takes Laura's time. So she can't focus on something else. So the traditional way in which we helped Laura was by writing RPA or robots. RPA are rigid rules based on rigid selectors, which were pre-written on a certain app version. More often than not, the app would get an update and the selectors would break, the rules would break, the UI would look different and nothing would work. So we had to fix it. Then the computer vision came. Now Otto could understand the screen, but he still couldn't understand the tasks. So the rules were still rigid. But we don't do only that. We also do agents. We actually, of course, we do agents, but we actually help our clients in building their own agents. Also, we help our clients in extracting every last bit of information from their documents. We call it document understanding. And we also do computer use. This is what I'm going to talk about right now. So Otto right now, the latest iteration of Otto, is a computer use agent. Of course, it's an agent. Always it's an agent. He now can see your screen, can emit actions as if he were you, and he can act intelligently. Otto is a React agent. He draws observations from screenshots, so he can see the screen just like you. He can understand what apps are on, and he can emit actions who are executed by our own driver. We call it a driver because it runs closely to the OS, and it actually simulates users' actions. People often say that a tool is only as good as its user, but our clients say otherwise. They will only use our tool if it's better than the user. So how do we measure the success? We use mainly 3 benchmarks. The first one is OS World. It's an end-to-end computer use benchmark. Then the 2 later ones are OS World G and ScreenSpot Pro. They are grounding benchmarks. I don't know if you are familiar with computer use grounding. It's about predicting the next action, the next click. Like I ask the model to save the file, then the model will predict where I need to click. Do I need to click in order to save that file? So it's sort of like a regression problem. But it's a very important regression problem, linear regression problem, but it's a very important linear regression problem because almost 80% of what we do on the computer is mouse related, either scroll, mouse move, or click. So that's why we need 2 benchmarks for it. Where do we start? So we start with open source model. We are not that big and we cannot really pre-train a model to do this, and it's actually wasteful. Which starts way below the human baseline. So the human baseline for your OS World is at 72%. It is heavily benchmarked, believe me, and it's like at 54% for OS World. When it's, I don't think it's intentional, but this benchmark is pretty old right now, and there is information about it all over the internet. And we need to get it better in order to close the gap between the human baseline and the model's performance. Our first intuition was, as always, to go with supervised fine-tuning. We have, being computer automation company. We have data sets with human-computer interaction doing tasks. But the problem with SFT and agents is that they usually tend to lack variance. They are usually a happy path. The model is usually overfits to them, and the real world performance isn't that great. We tried it. We got only 1 point as an advantage. But for the grounding task, it's actually all right because grounding is just one step. So it's mostly just like a linear regression problem. So it's good for it. Then we went to RL. Of course, we went to RL because it is what you need to train an agent. Actually, as you saw on the Tuesday courses, RL is asking an agent to go into a safe environment and do tasks. We put Otto in a sandbox and we ask him to do a task. We then score that task and update Otto's weights according to his performance. How do we measure his performance and update his weights? If you remember, we done the laboratory with the critic-based RL methods, which is a bit compute intensive. There are other ways. There are group-based methods, which are more data intensive, but less compute demanding. And I say data intensive, not that you need that many data, but you have to be careful which data you roll out, because otherwise you can have large groups with no variance and no updates. You'll see a bit later. So it gets a bit further than the SFT. For the grounding use case, the gain is not that much because the model has already learned that, which is great. In real life, the performance empirically, the performance is even better because the model is actually a bit more robust and resilient to outside errors. So RL is great, but there is no free lunch. The first complexity regarding RL in the industry is sandboxing. So you need a safe sandbox. You cannot really run the same computer use agent with other computer use agent in the same OS. So you have to provision and destroy your sandbox before and after the rollout. And this adds to the cost and complexity. And there is the rollout time. So for some tasks, the rollout time, the time taken to from the start to the end of the task, takes up to 85% of the training time. It's time in which you don't really update the model. You just wait for the rollout. A solution to this is the off-policy rollout. So I would say the classical way of doing RL is on policy. So you have a rollout, you compute the reward, and you update the model's weights. It's sort of like a synchronous programming paradigm where you have a lot of resources sitting idle. So you have idle GPU time, idle CPU time while you wait for the task to complete. The solution is off-policy rollout. So remember that for RL, we actually we actually have a term in the loss which penalizes the model to a model which goes far away from its frozen baseline. So it's safe to assume that you can actually do a rollout with an outdated model weights and then come up with the result, pass it to the updated model, and things will go similar enough in order for it to learn, and it greatly speeds up the inference and frees up resources. Actually, Frontier Labs are doing this in an extreme way. They have completely async rollouts and updates. Then another complexity is variance. But I told you that variance is a good thing. Actually, only the good kind of variance is good. Like we want the model, the policy, to explore its available states in order for it to learn what is good and what is bad. But we want to really control the environment in which the agent is running, because it's introducing some weird kind of variance, like we really want to control the pop-ups, the updates, and all the kinds of nasty stuff in the UI. We also want to control the app UI, and how it feels and the way it looks. For this, we devised the synthetic environment, which we call UI Cube, which mocks real-life applications. So this is actually our Concur and our Workday. And this is how it looks like in our mock version. It's pretty similar. It's not one-to-one, but it's good enough for our agent to actually learn something in a controlled fashion. The other main complexity, it's actually the worst one nowadays. Remember the OpenAI Hugging Face incident last week. It's trust and safety. There are actually real agents, actually even under-trained agents, which cannot really comply with your policy, which are running on real environments and doing real actions. So you have to really contain them inside an isolated sandbox, ideally without the internet, but you can do it without the internet. So you have to restrict the agent's mobility as much as possible, so it cannot escape the sandbox. So is the tool better than a user? I know I've showed you some results. Let's see what Frontier has to offer. So for OS World, the first version we are using, which was launched last year, I think, the results is yes, it's a bit better than the human baseline. Models like Mythos and Fable can easily achieve 85%, but also they tend to rely heavily on code. So they are coding models, and when you know how to do coding, everything becomes a code problem, which may be not that safe. But for OS World 2.0, which was launched earlier last month, the results are a bit disappointing. So OS World 2 introduces some long context jobs, real work cases, which the agents still struggle to achieve. We also, so the issue is I showed you a really low score compared to the human baseline. The truth is we are training a small model. We need speed. We need reliability. So we need to use, in order to alleviate this issue, we need to use a bigger planner model. So we have a model that plans and understands what are the tasks at hand, and we have a smaller model which is a shorter horizon model, which is a UI specialist. He never gets UI clicks wrong, but really struggles with long horizon tasks. So we devised this UiPath screenplay agent, which is actually a harness composed of a planner agent, a frontier planner agent, a large model which costs a lot and is really slow, with our quick and nimble model. And we actually achieved the #1 spot on OS World at the early January. And also, please observe how far we have advanced since January. So almost 20% on this benchmark. So I told you. So please take a look here. We have our model on our own benchmark, which is ours, but we share it with you. I encourage you to scan the QR code and take a look at our awesome benchmark done by our colleague Horia at UiPath. We only score 42 out of 90 with our model, which we call Path Mini. Gemini 3.5 Flash, which is a great model, it only scores 50 out of 90. But the combined harness with Gemini Flash as a planner and our own Path Mini as a agentic short-term agentic UI agent is really scoring 66 out of 90. And we can do it even better, I think, with more training. So there are still open problems left to solve. We still need to figure out how to do RL efficiently. We still need to figure out how to do rewards better. So we are also experimenting with PPO and critics. PPO, which is great for our use case because it allows us to actually critique real life trajectories. We also need a more rigorous evaluation harness. Right now, the OS World one is a bit too deterministic for computer. So for nowadays computer use. There are a lot of LLM judge based work done to evaluate agents. And we need all the kinds of work you can provide. So if you liked what you saw, please scan this QR code and get in touch with us. Also, if you see our colleagues, you can say hi to them and talk about, I don't know, computer use if you want, or document understanding or agents. They know everything. If there are any questions, please.
Thanks a lot, Radu, for the talk. Does anyone have any questions? Feel free to speak up if you're up for it.
Yes, we do process screen recordings. It's part of the process mining pipeline. I don't work there. I cannot give you many details, but yes, we do.
So if I understood correctly, you process the screen of your customers.
Yeah.
Where do you process that? On their computer, on your computer? Are you sending all this data? How do you make sure that, it doesn't leak?
Yeah, we have a zero data retention policy with our LLM providers. And actually the conversations are held on the user's computer. So we don't store any data. And we hope our providers don't store any data, and we have pretty good contracts ensuring that they don't store any data, they don't leak any data. We are actually very concerned about this because it's private data, I know.
What kind of headroom do you currently have? Like, what kind of examples are your models struggling with? And do you have any ideas how to solve those?
Yeah, actually, I can show you. I hope I have it ready. So just a second. Currently, we are training on really simple tasks like drag and drop tasks and I don't know, stuff you can do in Concur. But we will really want to extend to more real like scenarios and more varied ones because we usually automate. Thing is, we usually automate legacy apps, and they are a little bit fiddly, and we try to mimic those apps. So this is an example for I don't know, Jira-like UI. So this is really a task. It's a simple one. Just set the ML, I don't know, this ticket to done, and you are done with the task. So there are short and medium term tasks. This is how we train.
Thanks for the talk. I was just wondering what the context bloat looks like with these agents, like if they're running for hours at a time, processing everything on a screen. Is that like
Yeah,
Is that-
yeah, I got it. So for the agents we train, the LLMs we train, the small ones, the context is really small, like 30, 60K, because otherwise it gets really expensive at inference. Like you have to scale the KV cache and you have to have a lot more memory. But for the limitation is with the planar agents. The planar agents can go up to, I don't know, 1 million tokens in context. So they can, I think, run for half an hour without compaction, something like this. But the compaction is I think is the limitation where the context gets lost. So I think we can safely run for 50 to 30 minutes without losing any context.
Thank you for your talk. So you are having the model locally running. That's why you want efficiency you mentioned, right? And also you want a smaller model because it's also locally running. You want to optimize the memory. And I'm wondering, can you talk about this compromise that you need to make? Because is it the bottleneck now with the model because it's small, or is it the agentic workflow? And also, the results are great, if I may say, because you're running it locally, and then you have what 20% of the gap comparing with now 4.8 Opus, etc. So that's great. Can you talk about compromise that you're making?
Yeah, sure. So the compromise in the industry is that you need to make money. So you spend money to make money. We don't actually run the model locally. Even the 35B model is too much to handle for most of the consumer GPUs. But our clients could run the model locally. They can actually deploy it, and they are deploying it on their own on-premise data centers. And the compromise is that a bigger models needs more data. A bigger models needs more GPU, needs more memory, and it's not really that fast. And we've done that. We've used end-to-end large LLMs like Gemini 3 Pro with their computer use tool, but it's really slow. And the UX for the user, it's really bad because you see the model thinking for like I don't know, up to 30 seconds to do a click, and which is not really acceptable. So we are focusing on this niche, like quick and nimble models for computer use, which never do a mistake. We also try to train bigger models like planar models, but it's really expensive, and it really needs to justify the cost to do it.
Thank you for the talk. I have a question. I was looking in the part of the performance that you used, the part of Mythos. I think it was before or after the Fable 5, but I know that Mythos and Fable 5, they is quite similar, just that Mythos don't have a restriction like Fable 5. But in the field of performance, I know that Fable 5 is quite similar to Kimi 3, the Chinese AI model, and also you spend less tokens. So do you try with this, with Kimi 3 or Nemotron of NVIDIA?
Yeah, we are actually using Kimi. We love Kimi. Yeah. We are not limited to a single model for the planar model. We are limited to what our clients can work with. So we are really model agnostic. Only the model we train is the really minimum. Yeah, and actually I haven't included the Fable in this because it has a pretty similar score to Mythos, but they are self-reported scores from the technical report of the model. But yeah, Kimi, especially Kimi K3, it's really, really good, really strong.
We might have time for one more question if somebody's up for it. If not, let's thank the speaker.
Thank you.
And let's get some caffeine fix. Let's be back in 20 minutes. Yeah. See you soon.
All right. Hi, everyone. Hope you had a great coffee break. We will be starting the next lecture shortly. Please join us in the conference hall. All right. So as you can probably tell from our program, today is a day of exciting and emerging applications of artificial intelligence. And if you've been on Twitter at all in the last several weeks, you will probably know that by far the fastest emerging application of AI is in mathematics. And for this reason, I am particularly excited to welcome Matija Tapušković. Matija is a career development fellow in mathematics at Corpus Christi College at Oxford. And he is actually, his origins are from Montenegro. He has actually done his undergraduate education at the University of Montenegro. And he is a pure mathematician by training. So I think he experiences firsthand a lot of how these effects are changing the way mathematics is done. And before we start, I will just give you a fun anecdote. Both Matija and I were invited earlier this week for discussions with the president of Montenegro. And we were talking about, how to best prepare society for the emergence of artificial intelligence. And Matija actually had a really nice remark of, if you asked me 1 year ago if mathematics is safe, I would tell you mathematics is probably safe. But now, mathematics is very much exposed to this technology, and it's changing it in unexpected ways. So we look forward to discussing the implications and the recent exciting advancements with all of you today. Matija, whenever you're ready, the stage is yours.
Right. So, well, thank you very much, Petar. And thanks to all the organizers for putting together this amazing event and for inviting me to speak. I'll just fill in just a little more detail. It's a bit of a sort of full circle moment for me, this, because I've actually studied computer science as an undergraduate here in Montenegro, and then inspired by wanting to be able to read machine learning papers, I thought I'll do a master's in mathematics, and then I took it too far, as in Eastern Europe we tend to do. So, right. So I'm going to talk to you about ML for mathematics. So first, a disclaimer: I am a mathematician. My fields are number theory, geometry, and mathematical physics. I see this frontier of mathematics and AI becoming a little bit like what mathematics and physics has been for centuries. So problems and tools going in both directions. So if you imagine calculus was developed partly inspired by, wanting to describe the movements of planets and so on, and then end of 19th century, Einstein learns Riemannian geometry, early 20th century learns Riemannian geometry and develops general relativity, just uses it to model the gravity in the universe. So now we have this situation where mathematical tools in ML have become are being adopted more and more quickly. So you have things like Riemannian manifolds and diffusion models, sheaves in graph neural networks. You'll have resolution of singularities in mechanistic interpretability. This is singular learning theory. This is specifically from my field of mathematics. I believe that interpretability and formal verification are possibly the most important areas for maths and machine learning. But this talk is going to go the other way. So can machine learning discover new mathematics? That's the question. We'll have 3 strands in the talk, I hope, if we have enough time. One is learning from examples. Second, the first strand is learning from examples. So in a sense, classical machine learning applied to mathematical problems. Second will be applying LLMs, which is something that we more commonly think of applying AI in anything these days. And 3 will be this business of certainty and formalization and proofs, which might be new to some people. So this is about lean and so on. So in each case, we're going to discuss seminal results, understand when these methods kind of work, and discuss some challenges. In particular, I'll tell you about some problems that I have and why they're hard. Recurring question will be, where does the machine output re-enter mathematics? And I'll invite you to, don't worry too much about understanding each aspect of the mathematical problems, although that should be fun to do as well. And I've tried to make this very self-contained, but do try to notice how exactly where the models are in this story, because this is really an application story, right? So we need to kind of spot the machine learning. Okay, so hang on to your hats. We're going to discuss some math problems. Strand 1, learning from examples. And the strategy is predict, attribute, and then prove. This is the famous example that one of our organizers has been involved with, along with some people from the institute I work at. So this is about graphs and permutations. Permutations are, bijective maps from 1 to n to 1 to n, and we denote them like this, just by their image. And from a permutation, from a permutation group, we can construct a graph like this. So S3, this is all permutations of the numbers 1, 2, and 3, and you just connect by an edge two permutations if they differ by one flip. And then we call an interval between two permutations. For example, there in the picture 1, 2, 3, and 2, 3, 1, the whole subgraph between those two things. Okay. So in 1979, Kazdan and Lustig decided to each such interval, to each such subgraph, associate a polynomial. I'm not going to go into how, but it's a low degree polynomial compared to the size of the graph, it is very interesting in a field called representation theory. Basically, it's related to something called Schubert varieties. I'm happy to explain at some point what those are, but mathematicians really want to know about these things. So there's a famous conjecture called combinatorial invariance conjecture by Lustig and Dyer, saying that isomorphic unlabeled, so remove the labels, remove the 1, 2, 3, 2, 1, 3, and so on from that graph over there, and then you say isomorphic unlabeled interval graphs give the same polynomial. And so what Davis, Velishkovich, and others did and published in Nature in 21 was apply a GNN to this problem. So basically, just use a normal message passing GNN on an interval graph without labels, and predict ignore this picture for a second, predict the coefficients, and then predict the coefficients of the polynomial, right? Turns out you can do this with high accuracy. And so look at why. This is a standard technique in machine learning. So look at the gradients. If you remember the very first lecture of this summer school, you've learned about how gradients are used in training, and most of you know that already, but even if you're a master's student, this is sort of new. This is what I'm talking about. So gradients, you can sort of track, you can track through the training process, you can track what the gradients are doing, and you can therefore ascertain which sort of elements of the input graph in the GNN had the most to do with the eventual output, with the eventual classification of coefficients of the polynomial. And so what this does for you is in the initial graph, it flags some edges saying these are very important for this classification, right? So this is the mathematically new information. This is the mathematically new information. This is what mathematicians didn't spot, simply because these graphs grow incredibly quickly. As the number of, so this is S4. If it grows, if you try permutations of 5 numbers or 6 numbers, these things blow up very quickly. So you can't really look at them and get any intuition for them. So the GNN flags, the attribution method in the GNN flags these edges for you. And so you stare at it and you realize, or they realized, that you have, that they sort of split up the graph into 2 parts. On one hand here, we have a graph where this number 1 on the beginning is fixed. So that part is, you can just treat it as permutations of 2, 3, and 4. So this is a smaller problem. So suddenly you have a smaller problem plus something on the other side, some hypercube on the other side, and so on. This is very nice because we haven't seen it before. And so in a theorem, of the authors on the previous slide, sorry about that, including Veličković and Jordi Williamson, they then prove a recipe that computes the polynomial from a split of an interval into two smaller pieces. But this recipe of theirs needs the labels, which we don't want to solve our conjectures. So then they formulate a new conjecture, saying any valid split that you can construct, agrees on the polynomial. So that would imply combinatorial invariance. And in fact, a variant of that idea, so not directly this kind of split, but a variant of that idea led to a proof of the invariance conjecture in broad families, not in all cases, but in broad families by these two authors in last year. So this is a very successful example of where a machine learning model tells you and gives you some new mathematical information that leads to a new proof. Another problem. This one is sort of, from the machine learning perspective, so elementary that you will wonder, as everybody did, why did no one notice this? So an elliptic curve is an equation like this. It's one of the most studied equations in all of mathematics. A and B there are integers. It's related to elliptic curve cryptography and modular forms, Fermat's last theorem, all kinds of big mathematical ideas. And what mathematicians want is to solve these solutions using rational numbers. So which find rational numbers x and y that satisfy these equations. And they found that actually these solutions split into two parts, what we call the torsion part and the free part. It doesn't matter really. The left part, the torsion part, is very well understood. The right hand is one of the deepest problems in mathematics. So computing that number rank of E is one of the deepest problems in mathematics. So basically, we want to count the solutions of this equation. Now, turns out that's too hard. Nobody can do that. So instead, work in modular arithmetic. Look at this equation modulo a prime number. And so NPE there is the number of solutions of this equation in modulo a prime number, right? So modulo 3 solutions, 0 and 1 can only be solutions, and so on, modulo 5 and so on. So write down these numbers, AP, which is the prime plus 1 minus this P plus 1 is just a scaling, and represent the elliptic curve instead of the equation, represent it with this vector of APs, and then use this vector to predict the rank, right? That is what you can try to do. Data already exists. There is a huge database online that mathematicians have been tirelessly building for decades. So some people in 2022 literally applied logistic regression to a 500 dimension 500 vectors of these and tried to decide whether a curve should be rank 0 or 1. And they got accuracy over 97%, which was insane. Mathematicians didn't really notice that. They knew that you could predict it, just not with that high an accuracy. So experts said, oh no, they gave some technical thing. They sort of said, no, you just learned something trivial. You just don't know what you're talking about. This follows from the famous Birch-Swinnerton Dyer conjecture. We won't worry about it. So they ran another experiment, which which goes around this complaint of experts, and they got 99% accuracy. So what's going on? They gave it to an undergraduate student to tell them, can you tell me where's the signal? And he was sort of sleuthing away on his computer, wrote down these averages. So in this database, elliptic curves not accidentally, they're ordered by these numbers n. So each elliptic curve has an associated number big N called the conductor. It's a sort of arithmetic complexity measure of the elliptic curve. And so he took an interval of these numbers, so I don't know, 7,500 to 10,000, and he took all the elliptic curves there and then averaged a single entry in that vector, AP, averaged over the elliptic curve in a single interval of conductors, and plotted it and got this. Now, again, I cannot stress this enough. Some of the smartest people in the world have been looking at that equation and never saw this signal, right? Anyway, since then, Peter Sarnak, one of the great number theorists of the age at Princeton, gave his PhD student the task to try to figure this out. And she proved it in a certain class of modular forms, which are not quite elliptic curves, but are sort of related. And since then, people have been finding this signal in new classes of special functions that are important all over mathematics. So, I would call that low-hanging fruit. I don't know, for machine learning in math. Okay, so let's see where we are with the time. So, okay. I want to tell you about a problem that I have been thinking about for a long time, and maybe why it's difficult and why this applying machine learning to maths isn't directly, a cure-all for all of our problems. So what I'm interested in, sorry, let me just do this, are these things called Feynman graphs, which are graphs that look like this. They're basically graphs with some decorations, and they describe interactions of particles. They're sort of a bookkeeping device. To each graph, you can associate an integral. And that integral, is the integrand of the integral is just made up of graph polynomials. So basically something that you come up with, the polynomials that you come up with by counting some spanning trees in the graph. And so, okay, so a graph gives you the integral, and you sum by loop order. So you order all the graphs by how many loops they have, and you get something called the amplitude, which is the square root of the probability of a particle sort of event happening in a collider. Amplitudes lead to collider predictions, which are some of the most precisely tested numbers in science. There's a huge industry in computing these things, by the way. So goal, compute as many Feynman integrals as possible. Turns out that's very hard. So because it's very hard to do in general, we specialize to this kind of toy theory called phi to the 4, where only certain graph topologies appear and no masses and momenta. So, right, they look like this, like the ones on top. And here we have this situation that Feynman integrals carry a sort of complexity grade called the weight. It's generically related to the number of loops by this equation. And on rare graphs, this weight drops. So meaning the integral is less complex than we expect. So task, an ML task potentially, detect the weight drop. Okay, so this turns out to be very hard. So we, again, do something similar to the murmuration setup, and this was completely unrelated, by the way, to the other story. Count the solutions of, sorry, this phi should be u, actually. That's my mistake. So this is u of g from the previous slide. So again, count some solutions modulo p, and put these in a vector. Like before, we had this vector of APs. This is now a vector of these things. We call this vector C2. Conjecture says, the famous conjecture says that if C2 is a zero vector, it's a zero vector if and only if the weight drop exists or if the integral is simpler. Okay, so, let's apply the same idea. Is weight drop visible in elementary graph features? I took you take around, I mean, these numbers don't really matter because the data is very scarce. This is my point, which is the next slide. But basically, if you take about 2,000 graphs, try to do some classifications, you get pretty good accuracy. Top feature, but it turns out that the top features, if you then try to attribute, this is not the GNN, by the way. Top feature just restates a theorem that we already know. So yeah, something called the tree vertex cut exists, leads to a product, leads to C2 being 0. This is known. So let's remove these positives. You get this number. And but it turns out that actually all of these 2,000 graphs by known theorems classify into 165 families, of which only 7 have this weight drop. And basically what this is doing is learning when the sort of the labels are constant within a family. So you need to recognize this guy's siblings. The theorem exists for that. So if you hold out this, now this drops significantly. Okay, so we get some previously known results, nothing too interesting. Now, this is my main point of this. Data issues. It's very hard to compute all of those things. We only have in phi to the 4 about this many irreducible families. So after we apply all the known results, we only get about 283 graphs. catalog, which exactly 7 have weight drop. Now, these 7 are conspicuous. They're much more symmetrical, and they have spectra of a particular kind, but there's just too few of them to conclude anything new. It is conjecture shaped, though. Exceptional weight drop lives on exceptional symmetry, and so on. Now I want to tell you why this is expensive, right? At 11 loops, that C2 vector is known only to 4 primes. The next prime can cost hours per graph to compute. Why wouldn't you, now even if we had data, it would be trouble trying to apply at least MPNNs, because 5 to the 4 are by definition 4 regular graphs. And as a lot of you will know, MPNNs are blind exactly to the global signal that would work because of this 1WL constraint. The fixes do exist in the geometric DL toolkit, but we still have the data problem. Now, alternatively, we can consider the real decorated Feynman graphs, not the toy theory, but the sort of real physics. And then the problem is that a single integral can be a whole paper to just compute a single integral. They're just too hard. Now, I do want to say that I have been working on a theory that sort of makes this problem into something tractable. And that's this idea that behind every invariant there is one object. That object is called the Grass-Mellin motive. For those who have studied some things with sheaves will know that what a vector bundle with a flat connection is, and this thing in one of its guises is a vector bundle with a flat connection. And what you're actually looking for when you're looking for weight drop is a fiber of this vector bundle simplifying. Okay, this is just an aside, ignore that. Now, this is more interesting. These Mellin motives these objects associated to Feynman graphs are representations of a group, which we call the cosmic Galois group. And then the question is, can this action be learned? And some people tried. Transforming the bootstrap, this is an interesting paper in a different toy theory called N equals 4 super Young-Mills by Kai et al. One of the main authors of the paper, Lance Dixon, tried to explain to me what they managed and what they didn't manage. He was not too excited, but there are some results. We were sort of trying to survive hiking on a Swiss mountain, following 2 colleagues who were much more experienced hikers than us. So I don't know, maybe that had something to do with his lack of enthusiasm. Anyway, but there is some work on it. Okay. Let's do something about. Let's talk about alpha evolve, given the context of DeepMind. So in mathematics, often you don't have a data set, and I'll give you some examples of this. You don't have a data set. You just have a way to say, okay, this is a proof of something, or it's not a proof of something. But of course, as all RL people know, if you can score something, you can go around searching for things. Key idea that people at DeepMind had is don't search the space of objects, search the space of programs that generate objects, right? So to start, write in Python an evaluator for an object, and then a program that generates objects, a very simple one, possibly a dumb one, and then give it to an LLM. So a program score is the score of the object it generates, and an LLM will propose for you from a starting program a handful of similar programs. And then evolutionarily, it sort of just goes on searching for the, sort of putting together the best ones and so on. The strategies are quite clever. I'm not going to talk about them. This goes back to AlphaZero and all of this stuff. So the hope is that the objects we care about have short descriptions which can then be understood from a Python program. So basically, that by the end of the search, you find something that satisfies has the score you want, and you can transfer it back from Python into mathematics. The history of the idea briefly: Wagner in 21 did extremal combinatorics with deep cross-entropy search. Then Mehrabian and others, including Adam and Peter again, did extremal graphs with AlphaZero. And then there is these 3 ideas and ending up with alpha evolve, which is the current sort of industrial scale implementation of this idea. The main problem, the main sort of flagship paper of this is Georgiev, Gomes-Serrano, Tao, and Wagner from late last year. They tested this on 67 mathematical problems for various areas, number theory, geometry, and so on. Nearly all of these problems are sort of improve abound on a constant type problems, and the scorecard they came up with was this: 20 new results, 39 matched literature, and 8 fell short. So just to sort of deflate things a little bit, and in the author's own words, none of these results are individually important. Every one of them could have been found with usual tools, but the problem is time. You just don't have enough human time to sort of work all of these problems out using standard tools. Giving it to a machine is much better if that can be done in some sensible way. And Tao says that the tool beats the literature where the literature is thin and matches it or trails it where the literature on the problem is deep. And he also uses this term long tail problems, which is sort of, you know, problems that you might give master students or PhD students. So I really like this for a particular reason, that it will force PhD advisors to give students more interesting problems to work on. Okay, so yeah, another opinion. The expertise moved into the evaluator. I'll show you why on my own problem later. Okay, so this is the most famous two problems they worked on and where they got most results. This is a bit technical, so apologies. The arena you work in is this finite fields with prime power elements in it, and this is three-dimensional. So imagine sort of a lattice in 3D, which is sort of Q, it's Q integers long in each dimension, and all dimensions wrap around. So because you are working in modular arithmetic. And then the question is, a Kekia set is a set where a line in every direction in your space exists. I'll leave it to you to imagine that. A Nicodim set is a line through every point, excluding has to exist in the set and excluding the point itself. And then the question is, find the smallest such set. How many points can you delete and still have these properties here? Previous records for Kekia in 3 dimensions is this number with this of p here. Sorry, this should be small p. And AlphaEvolve improved this when working with primes which are 1 modulo 4. Basically, it sharpened this here to 1/8. A 2-line formula. They came up with a 2-line formula, an algebraic formula over quadratic residues that DeepThink then proved is correct. A previous record for the Nicodim sets was literally random chance. Delete random points at a fixed rate of deletion and you get this bound. But AlphaEvolve had the idea, don't delete points at random, delete algebraic surfaces. So delete anything that lies on a quadric or something like that, on a sphere and so on. But as used, this was no better than random. So AlphaEvolve couldn't quite get there, and Tao had to repair this. Could repair this by hand and obtained a better bound than this. So this is another example of, you know, it spotted something mathematicians didn't think of, and then a very good mathematician, albeit in this case, repaired the argument into something that works. Okay, I'm going to skip these because we don't have time. We'll come back actually to those problems at the very end. Okay, so different possible outcomes of the search trend. A record with no structure. Sorry, I didn't talk to you about autoconvolutions, but this is one thing where you just get a number out of it. So just sort of a better bound on something, and you don't really see any new mathematics, particularly in it. Full construction and a formalized proof. This is Kekia in D = 3. I'll explain to you this thing about formalizing proofs later. But this is kind of the holy grail. You just give the problem and an evaluator to AlphaProof, and it comes up with an argument. Well, it comes up with a Python program that DeepThink then converts into an argument, and then AlphaProof will convert it into a formalized proof. But this only works for simple enough proofs, it turns out, for now. Now, okay. Another option is a full construction and an informal proof. So, you know, DeepThink does come up with something that humans have to check by hand, basically. And then there is the third option, which is a seed of an idea. This is the same as in the first problem in strand 1 that we worked on, that we talked about, which is the GNMs and intervals, Bruhat intervals. So here we have these Nicodim algebraic idea that Tao had to complete. So we have 2 diagnostics for when we can apply this. Does the object have a short description? We can't know that until we try. And is the score an exact cheap count? So in all of those problems, the scoring, the evaluator Python program was milliseconds per instance. So you could test out many different ones. Okay, so I want to tell you about the problem I think about. And again, why it's very hard. So zeta function, this is the Riemann zeta function. Looks like this. Very classical, you know, the Riemann hypothesis and all this business. Even values have been understood since Euler. So this is the value of zeta 2 is pi squared over 6, and every even zeta value is a rational multiple of this. Now, odd zeta values are very, very mysterious things. So zeta 3, zeta 5, and so on. All that is known about them is this. Apéry in 1978 proved that zeta 3 is irrational. Balrivois in 2000 showed that infinitely many of them are irrational, and Zudelin proved that at least one of these 4 is irrational. But if I ask you, is zeta 5 an irrational or a rational number? No mathematician can tell you that. It's a very, very big open problem. Now, how did Apéry prove this? Just to get some feeling for the problem, he found a series, an infinite series of fractions like this that approach zeta 3 abnormally fast. So because fractions naturally repel each other, if this theta here is over 1, this is a contradiction. So you can't have, this leads to a contradiction. Zeta 3 has to be irrational. The fractions come from, in Apéry's work, these fractions come from a miracle recurrence relation that nobody quite understood where it came from, and he didn't tell anyone, which is a standard problem of mathematicians. Baukers in next year, sort of 1979, said wrote down a family of integrals like this with these dials A, B, C, D, minus E. So these are like integers. And he said, when you put them all off, except the last one, you get zeta 2. So I'm working with zeta 2. Zeta 3 is one dimension up, and the same thing works. And if you make a choice, if you take I of n and n plus 1, you get this type of approximation that Apéry. So this is sort of Qn over Pn from the previous slide. And so zeta 3 is the same machine, one dimension up, but it has 9 dials, not 5. Now, Francis Brown, a colleague of mine, my former PhD advisor, recently explained how these can be viewed as periods of Mellin motifs, the thing that we worked on together, the same objects that we had in strand 1. Now, this actually doesn't solve the problem for us, but it tells us why it's hard. So a candidate proof is therefore array in this dial space, and the proof lives on the locus where the underlying family of integrals degenerates, similar to Feynman integrals. Dial counts are a problem, though. Zeta 2, we have 5. Zeta 3, we have 9. Zeta 5, we already have 20. So this is hopelessly large. A scoreboard that we have for now, zeta 3, Apéry got 108, and zeta 5, we need more than 1, and we have 0.86. Strand 2 shape, exactly, but only if we can score these things. The problem is, to score 1 point in dial space, we must compute this theta. And that's difficult. So there are 3 rungs to this computation. Rung 1 is error decay, which is a numerical computation that's very quick. And in geometric sense, in this Mellin motif sense, in this geometric view, this is a differential signature of the space, and that's of the vector bundle, and that's easy to work with. Rung 2, though, takes minutes per instance. So you need to compute that recurrence. And that's a symbolic computation, or in geometric, there's a geometric view of this as well, and that's expensive too. And rung 3, well, it's not even a computation. So theta is an asymptotic number. You can compute, so obviously you can finitely compute for the 1,000 QNs or something. But this rests on sort of massive hidden cancellation that you have to prove for infinitely many n. And so but still, if these 2 fired on some sort of AlphaEvol program, then at least we would know where to look. In the 67 problems that I described, that people from DeepMind and Terence Tao and Jody Williams worked on, was the evaluator was cheap, exact, and it was crucially ungameable. So the RL couldn't sort of find some clever way to get around the real problem. Here, search is not the bottleneck. The evaluator, building the evaluator, is the actual research. Okay, so we know we did some calibration. We know that the haystack is real, but we know which of the properties of the needle actually admit a metal detector. So, some progress. Both here and in finite graphs, the signal we're trying to learn is the degeneration of these geometric objects called Mellin motifs. Okay. Finally, we want to discuss formalization. This is the most, I think, active currently area of AI applications in mathematics. So let's start at the beginning. What is a proof assistant? So this is a new stuff about programming languages. I hope it's sort of, this makes more sense. Lean is the most famous one. The idea is you write down definitions in a programming language. You write statements of theorems in a programming language as programs. And then the kernel checks that you can get from one to the other, the kernel of the programming language. So you build statements up until you reach the theorems you care about. I'll give you an example, a trivial example in a second. Compiling a statement, this is a sort of type checking situation. Compiling a statement checks if it is well formed, not if it's true. So a statement is a type, and a proof is a term of that type. And you have to construct these terms. Here's an example. So it looks scary, but it really isn't. So the input of this toy theorem is 3 natural numbers, A, B, C. And we have 2 hypotheses. One is that A is equal to B, the other is B is equal to C. And this is the statement after the colon here, the semicolon here, A plus C equals B plus B. And after this, after assignment, this is where the proof starts. And this by just starts an interactive version of this, but in the terminal. So, what this command does, it says rewrite this expression using hypothesis 1. So it scans from left to right and goes replace A with B. Okay. So now we have B plus C equals B plus B. And then it says rewrite, and then you say rewrite using H2. So it goes from left to right and finds B, replaces with C, finds these 2 B's, replaces them with C, and you have C plus C equals C plus C. So that's true. Similarly, you could have something like this. We have an AND construction here. N plus 0 equals M, and M plus 0 equals N, if N and M, hypothesis N and M are equal. Constructor tells you separate the AND into 2 different things to prove, N plus 0 equals M and M plus 0 equals N. And again, you just apply rewrite at 0 means N plus 0 gets, it replaces M by M plus 0. And it replaces M plus 0 by N, and then exact H says apply this hypothesis, and that's true. And then for this one, it's the same thing, but sim flips the order. So anyway, that's how proofs in Lean look like. Why would you want to do this? Well, would you read a 150-page proof of a major conjecture handed to you by an LLM? Would you read anything in any field if you're an expert in the field and your postdoc gives you 150 pages generated by Fable? Is that a good use of time? No. So that's the idea. The main source of this, the main source of current sort of the storage of all current work is this library called MATLAB. It has 2.3 million lines of code, took about 9 years to complete, roughly contains an undergraduate degree in mathematics, formalized, plus a jagged coverage of research mathematics beyond that. So, statements of open problems can be found, for example, in DeepMind's formal conjectures repo. Once humans agree the Lean statement captures the conjecture, checking any claim disproof should be trivial. If you have the proof, you have the conjecture. The problem is it's very tricky to actually write down a conjecture in Lean and have it mean what mathematicians think it means. This is a general problem with machine learning, with AI. There's this thing of problem of meaning. It's been sort of an undercurrent of a lot of talks in this representations and so on. Okay, so here's the history. So for a baseline, this is sort of IO International Olympiad in Mathematics business. So just to see what it does on very hard high school problems, where we are with this stuff. In 2024, DeepMind used alpha proof and alpha geometry to sort of win a silver medal. This is an RL sort of method using Lean proofs as the kernel of Lean as an evaluator. And it could solve one problem in minutes using this, and then took 3 days to solve the others. to the standard it did, to the silver level standard. But at least it was formalized, right? In 25, they dropped the lean and they just work with DeepThink, which just worked in English, and it worked within contest hours. So I don't know, 5 hours, however much that is. But it had to be graded by humans, and it was, again, this is very good. This was gold standard, but again, wouldn't necessarily work for research mathematics. This year, a few weeks ago, Axiom Math used their Axiom Prover to work again in lean and win gold. Problem, we are back to 25 hours of working time, right? So you see, you see the problem, right? Hard problems, lots of compute, impossible to check by hand, or a waste of time, depends on how you think of it. Or, you know, ideas and so on, good ideas, but, you know, less time, but then you have to do sort of human work to make up for the thing. Okay, so back to the Kakeya problem that I mentioned before. So AlphaEvolve, I mentioned, found this construction for a Kakeya set in 3 dimensions for p equals 1 modulo 4. DeepThink wrote down an informal proof like a mathematician would. And then AlphaProof converted it into a lean proof, which compiled. And this was the full pipeline closed. However, this is kind of a humble problem. So in fact, also last week, weirdly, for work on Kakeya sets in Euclidean space, in sort of RN, another field medal was awarded to Hong Wang. A very different problem from this, much harder. This sort of a toy version of that. The paper's caveat, this is in the paper of Tao, Wagner, and others. This only worked because every ingredient was already in MATLAB. Now, in dimension 4, interestingly, elliptic curves appeared in the DeepThink argument. So it said, remember elliptic curves from strand 1? So it said, here's a construction for a minimal Kakeya set in this dimension, and for these primes. But, you know, it has something to do with elliptic curves. And AlphaProof just said, well, you know, can't help you. So humans had to check this by hand. And for Nicodim sets, AlphaEvolve's construction was too tangled, even for DeepThink to analyze. And final proof was by Tao. So, you know, just some sort of scaling of how hard things are. Now, the frontier of machine certification runs out exactly where deep objects come in. That's sort of the message of this. But the frontier is moving. Okay, so how does this scale? Does this sort of formalization stuff scale? It took humans about 6 months to check in lean a very hard theorem in condensed mathematics that Peter Scholze, one of the cleverest people alive, wrote, but wasn't sure if it's true. And he asked some people to try to prove it in lean, and they did. And in fact, they simplified his proof. So, but it took 6 months, just to give you a scale for how long people take to do these things. Now, for Maslow's theorem, this is the famous statement proved by Wiles and Taylor modularity theorem. Via the modularity theorem, Kevin Buzzard at Imperial got a 5-year UK government grant running to 29, planned with, so he planned the route with Richard Taylor, one of the original authors of the original theorem. And they're still not done trying to formalize this stuff. It covers one branch of arithmetic geometry. It's a very important branch, but it's still one. Now, in July, again, a few weeks ago, the modularity lifting theorem, a part of this modularity theorem has been finished and formalized using, I think, Fable or Sol or whatever, in 250,000 lines of code in 2 weeks, supervised by a PhD student of Buzzard's. Okay, and in most mathematics, including some that I care about, we have no mathematics at all. So if anybody wants to help. Anyway, so here's another example. Wiazowska won in 2017 Wiazowska in 2017 proved a very important, very interesting theorem that she won a Fields Medal for, called the sphere packing theorem. Humans wrote the definitions behind it and the blueprint for the proof. But then Gauss of MathInc, Gauss, a formalization engine, finished the dimension 8 sort of proof sort of just as an LLM in about 5 days. And in 24 dimensions, the second sort of higher dimensional statement of the same theorem, they needed 2 weeks. And but we don't know the plan. We don't know how much it cost. We don't know anything. Basically, we know these. I mean, 2 weeks doesn't mean anything if they don't tell you how much compute they spent, right? So it generated 500. This is for the smaller dimension. 500,000 lines of lean code. Remember, 2.3 million in 9 years so far we've had, and they generated this many in 5 days to prove this one theorem. Now. Right, so this relates to sort of trust economics. Read the 30-line statement of the theorem, trust the kernel of lean, nobody refers the thousands of lines in the middle. Now, why did this work for this problem and not for Fermat's last theorem? Why does he need a few million pounds from the British government, and this was done by a software from a company in 5 days? Well, sphere packing's definitional tower is very shallow. Fermat's last theorem is just mind-blowing. So, okay, so sphere packing, I can tell you what it is, right? I can tell you the statement now and you'll understand it. It's literally this: take unit spheres, take an 8-dimensional real vector space, and I tell you, tell me the points at which to put the centers of those spheres in R8 so that the spheres encompass as much volume as possible, right? Simple. Fermat's last theorem, Fermat's last theorem is, I can tell you the statement of the modularity theorem, which implies Fermat's last theorem. So it says that every elliptic curve is modular. Simple enough. What does modular mean? Well, modular means that L functions of elliptic curves and a modular form agree. What are L functions? And so on, right? It takes decades, hundreds of hours of graduate courses to understand this. Okay, so formalizing proofs is accelerating, as we've seen, but definitions are the hard part. You really need to get all the definitions correctly, and people have to agree on them before you can trust anything. And this has to do with this sort of compressed meaning situation. So we really need, mathematics is, I heard once a great mathematician say that mathematics is about condensing as much truth into as small a statement as possible. And this is very much related to that. And this was 10 years ago that I heard this. So yeah, okay, before any of this business. Okay, so in, just 3 counterexamples, because we just about have a little bit of time. I went a bit quicker than I intended to, which is good, I suppose. Right, so again, some counterexamples. This seems to be where LLMs excel. Proofs, we're still, not quite sure about how to go about that. But counterexamples are just, blowing up. So we had 20 May, 11 July, 19 July. Erdős unidistant conjecture, this proved by an LLM. This is sort of connected. What it did is connect this problem with a problem in a completely different area of maths. But, this was all very well documented. Literature was there. And then it, because people wanted to understand if it's correct, they decided to formalize it instead of just trying to read it. And they formalized the axioms in 5 weeks. 5 weeks later, they formalized everything and they got 1.2 million lines of lean code. Oh, sorry. This is 3 weeks is the proof, and axioms took this long. On 11th of July, Grothendieck's 60-year-old group scheme question got a counterexample and auto-formalized in 4 hours. But this was much shorter, 1,100 lines of code. This is extremely exciting for me because this is my area of maths. It's about group schemes. I don't want to bother you with it. But so far, I've been sort of sitting around thinking, why is everybody working on Erdős's problems? I mean, I understand why. It's because definitions are very, there are very few of them, and you can very quickly get started on a computer with them. Grothendieck's work takes a long time. But yeah, still, they're managing to do stuff. LLMs are managing to do stuff there. Now, on the 19th of July, this is a famous one on Twitter now. The Jacobian conjecture, which was open for 100 years, was found. The counterexample was found. The verification here is trivial. It's literally, the conjecture says if you have a map from C3 to C3, complex 3 space to complex 3 space, it's a polynomial map with determinant non-zero, does it have an inverse? So it's sort of local. If a map is locally invertible on a particular kind of very simple space, so C3, if a polynomial map is locally invertible, is it globally invertible? So, is it surjective or injective? And it turned out, Fable, I think, found a counterexample. Problem, again, we don't really know. We're not given the prompts. We don't know how it was done. But it's exciting that it's there. I wouldn't assign too much value to the 100 years there or to the number of these things, because to me, actually, I think that what we now need to do is understand the meaning behind these counterexamples. And if we can do that, then we've created some new mathematics. Because what mathematics is about humans understanding new things that are not intuitive necessarily by just looking at the world. So this is, why develop complex analysis or things like that? Why quantum mechanics, tools for quantum mechanics and so on? Because we can't just look at subatomic particles and think this is how they work and let's put things together and make a computer or whatever. So we really need mathematics to understand things. And this is, so humans need to understand these counterexamples mathematically and then explain it to other humans. That's when we have new mathematics. But still, this is exciting stuff. I'm just hoping that you got a little bit more of an idea of why mathematical problems are subtle. It's not all about just, here's a theorem, example, counterexample, and so on. It's really about understanding and being able to convince others that what you have done can be trusted. And this is something that mathematics and machine learning in mathematics has in common with machine learning in every other field and AI generally. We really need tools to be able to trust the output and to be able to extract meaning from the output. There's a very nice quote from William Thurston around this subject. So I'll stop there. Thank you.
All right. Thank you very much, Mattia. That was very exciting, and I believe a start of what's to come, like really in this field and beyond. I will start, before opening the floor to questions, I will start by asking one which I found very curious about the Jacobian conjecture. Like, I saw quite a few statements floating around that the counterexample was so overlooked because a lot of the people were trying to prove it's true rather than trying to look if it's false. Do you agree with this, or do you think we missed it for a different reason?
I don't know. I don't know. I haven't. I've heard people told me that they have. I've seen. I know that people have worked on it sort of on the side occasionally. I'm sure that both angles were explored. I'm pretty sure that both angles were explored. That sounds a little bit like a defensive argument that, mathematicians didn't find it because we weren't actually looking and so on. This seems to happen a lot these days, although it's sort of slowing down. People are accepting that we have to work with this stuff. It's actually very interesting. So my guess would be, that's just some people. I don't know. There was a very interesting blog post, from last week of Tao, explaining sort of doing exactly what I just said, digesting the counterexample.
Yeah.
And what it does is interesting. It explains, to me at least, it explains why the LLM didn't do a stupid search. It kind of did something along the ideas of seems to have done something, could have done something along what Alpha Evolve does. It sort of searched for search strategies and then found a framing of the problem where the search is much lower dimensional and then found the counterexample there. So I think that's, and to do that sort of, that angle, there is literature on that. So it could have, I don't know, maybe it implemented something like that.
Okay. Cool. All right. Any questions from the audience? Yes, what?
Thank you for the nice lecture. So my question is, what do you know what kind of LLMs were used to generate these lean proofs outside of the Google people? Like,
Yeah, just okay, so Axiom has its own, I don't know what they're using as a base, for example, but they're building their own, they have their own engine, just a formalization engine. Same with Gauss. So those are the MathThink people. So I'm not completely sure what their sort of background LLM is. These counterexamples, Jacobians and so on, those are Fable and Sol. The one in Fermat's last theorem that I said a PhD student used to auto-formalize a big theorem in arithmetic geometry, that was at a workshop where both ChatGPT gave access to Bazard and his people to solve to the latest Sol version for free for unlimited amounts or whatever for a week, and then Anthropic jumped in and said no take here have some Fable and so on, and they used both. So I don't know exactly which part is which or whatever. It's a huge amount of code.
yeah, I was curious, how does it get the context of the MATLAB library? Like, but I guess if it's some proprietary, like Axiom, they have they train their own LLM, so you don't know what are they doing. Okay.
Yeah, exactly.
Thank you.
Any other questions?
So I wonder your opinion. Some people say that I'm not mathematician by background, but they say that actually coming up with conjecture is much more important for mathematics than proving it. So can you comment on that?
Yeah, but okay. But basically, I disagree with that. Actually, it's not. I don't even disagree or agree or disagree. I think that philosophical questions have the only worth of philosophical questions to me is, to keep me inspired. So I'll have a philosophical debate after a drink in the evening before the next day I get up, pumped to do some work. But this is. You can't, I mean, I think this is kind of the direction of this is proving that proofs are important. Trust is important. Meaning is important. That's not contained in conjectures. Conjectures are just musings, which are also important, right? We don't live our lives reading sort of only nonfiction. We read both, but we read fiction and nonfiction to make our lives nice, right? So this is sort of a similar relationship, I think. And yeah, so for formal mathematics really matters. Yeah, conjectures are what drives the field somewhat, but actually conjectures come usually from proofs from previous work. So people try to prove something and then find a reformulation. Instead of proving it, they find a reformulation of the problem, and now the problem looks different, and now that's a new conjecture. This is exactly what happened in strand 1, the conjecture that Petar made along with his colleagues, right? So that's great, but the real work is how you get from the previous statement of a conjecture to the new one, and then to the actual proof. That's my opinion.
I see there's a few questions. Maybe let's start.
Thank you very much. Can you comment how important it is, and if you have observed not just at, let's say, of course, new proofs that we observe daily, but new approaches or angles to tackle the proofs, and how the LLMs will help you finding the new approaches to prove something. In all these lines of lean that we are observing, are there some ways that will be breakthroughs in how we approach tackle proofs?
Right. If I understand the question correctly, it's like lean doesn't help us gain intuition. Lean is about trust. So intuition comes from, maybe deep think can help you get some new intuition by scanning a vast amount of literature that you can't scan yourself. Possibly. It's not clear to me that LLMs are better at that than human, even though they came up with these counterexamples that we haven't. It's not clear to me, we would have to have a control sample of how much time people spent on it versus LLMs and all this kind of stuff. I have no idea. So whether LLMs are good at actually providing brand new ideas without guidance, I don't know. Maybe they will be. But auto-formalization, this stuff where an LLM translates a human proof or an informal proof into a formal one, that's just about us being able to say, okay, we're sure this is correct, even though it was generated maybe partly by an LLM, right? That's a completely different question, extremely important, I think.
Thank you for the talk. So, like, if we try to classify the unsolved problems that are just out there, like close to our reach, we can think of the problems that are analogous to something that has been solved already, and you would expect that LLMs would perform well. But what about problems that would require either applying something, some kind of unusual information, like unusual lines of reasoning or something, or the problems that would theoretically be solved by just thinking in a completely different way that like than the one that we tend to think about them?
Right. I think I mean, again, very hard question. Are humans, is this sort of serendipity in science important? Is there something special about the human brain where we're able to jump around in the search space in ways that machines can't? I don't know that. The Erdős unit distance counterexample did involve looking in a completely different part of mathematics. I would have to do some mechanistic interpretability work on that particular run to be able to tell you, why it got there. Did humans get there before? I don't know. Which path through the literature it took? No idea. So yeah, that's a very good question. It's a very hard question, but it's a very sort of philosophical, fundamental one that I don't think we will be able to answer. So because humans do that as well. Occasionally, a human makes a jump like that. And I don't see why an LLM wouldn't. So, yeah.
Thanks.
Yeah. Thank you much for the very interesting talk. I wanted to ask maybe a similar question, like, did we in any of these proofs observe like some super new kind of mathematics? Like, for example, in the past, we have developed like, all these linear algebra tools which we can then use to prove things outside of this scope, or like complex numbers or analysis and all of this, right? So did we see something similar like this, or it is just too difficult to parse from the lean proofs? Or
again, lean proofs are not where to look for that. It would be in the informal proofs generated by the regular, human language LLMs. But the answer is no. And this is fascinating to me. This is about, again, I'm as an algebraist rather than an analytic mathematician, I think about, structure, exactly that. So the Mellin motives business that I sort of mentioned, this is a theory that me and my colleagues developed to look at a problem from another angle. Now it relied on extending an existing theory, and and so it relied on existing tools. But it's about, here's a new definition, here's a property of that object that we've defined, and here's how you can use it to look at this problem. No, an LLM has not done that in any of these, right? Whether it would, whether we should guide it to do that, or I don't know, I have no idea. But for now, no, there's no theory building by LLMs that I've seen.
Thank you.
We have time maybe for a couple more questions. Okay, there's one here.
Thanks for the talk. I want to ask you what's your thinking about like LLMs will shape like the future research in math and in physics, because as I'm seeing like nowadays we're looking that there are some things that are like not worth pursuing that many times. No, like it's not worth doing a 6 years PhD in something that Cloud can solve in a week. So it may be like some way of new reshaping on how we tackle problems as humans, as we should spare more time in having like these insights as relativity or something like that, instead of just like solving gaps between theories that maybe are not worth time doing.
Right. If I understand the question, I think this is a question for everybody in every field, right? What's worth doing now, given this, new tools, and well, I'm just my thinking about this is it will. I mean, I know mathematical problems that are too hard, that LLMs are not giving anything on, and any of the tools that although I don't have access to the I don't have access to the really cutting edge ones, but they're not really giving us a huge amount of new ideas in these. And I think if you give an ambitious enough problem to, you know, the next student, the next postdoc, and so on, it will, you know, they will have work to do, even if they use LLMs. And even more than that, as I said, like understanding and sort of properly understanding, explaining problems to other humans and solutions, possibly building new theory to explain things and so on. These are things that are not gone. Like, I don't know if you ever tried to run Claude Code for a very long time and then have it explain to you what it did. This is not useful to human beings. And not because it's wrong, it could be right, but this is just not how we talk and how we understand things. So, so there is work to do. You just, I think, when I talk to my colleagues, I have had colleagues, oh, I don't know what I'm going to do to, you know, my whole work is in statistics and applications of statistics to something. And I'm like, how do I, you know, I'm improving algorithms and Claude Code is better at improving algorithms than me. So what do I do? Give PhD students? They can't follow my trajectory. It's like, okay, yeah, has the problem of, you know, mapping phenotypes to genotypes to phenotypes been solved? No. There you go, right? So this is I think the way to go. Just be ambitious. Yeah.
One last question. Okay.
Thank you for the talk. When you were mentioning DeepThink, you mentioned that its benefit is the speed, but then you need human verification. So my question is, how difficult would it be to implement like an automated system that would take DeepThink's output and construct like a link proof? Where is the main challenges there?
This is exactly what they did. This is what AlphaProof did, right? Took so this is the Kakea example that I gave. This is exactly what they did. I don't know the exact mechanics of it, but it's the same as if I wrote the proof and gave it to an auto formalization engine. It will just read it and try to build, you know, it's like coding. So yeah, it's just harder because informal. It's hard to describe this without, you know, sitting down and showing you a more complex proof, so you can really see why it's very complicated. It's very different to saying, you know, write a front end for a website or something, because of the sort of condensed meaning that I was talking about. It's very, you know, every sentence in a mathematical proof is very subtle and it has a lot of weight behind every word. So basically, you know, conveying this to the, you know, for an LLM to understand this and convert it into code is a very hard task.
Thank you very much for all the questions. Let's thank Mattia again. And really an exciting field to watch in the coming, well, even weeks, but also months and years. Right, so this concludes our lecture blocks for today. We now have our usual lunch slot, after which the free activity, the trip to Lovćen will begin. Milutin already shared with you all of the details about this at the start of the day. If you have any issues, please feel free to ask us on Slack. Yeah, looking forward to seeing you there. Thank you.